On site. It looks like sediment on top of a possible rock wall or rock ledge.
Mm-hmm.
We'll get closer and find out. We're about 15 meters above bottom. We got somebody down there at 6 o'clock. Maybe a sea cucumber.
Seems like a reasonable guess.
He's a little too far away for me to tell. It looks like the sand is really smooth, bioturbation in it, but no rippling. Here's an interesting bioturbated. Looks almost circular there, right at the tip of the temperature.
Oh, like something's been
spinning
from
around
a central area.
Yeah, radial.
Reaching out, feeding you.
Oh yeah, could be that too. Worms. Some infauna.
I want to know what that is.
I think it's a
cuke?
A cucumber, yeah, but I mean, it could be a worm. I have no, like, the distortion, like, I'm really disorienting if I don't have my lasers on. I don't know how far away from bottom we are. You know, it's like my brain doesn't want to, there's no reference point.
Hard to put it all together.
Yeah.
You mean that purple thing in the distance? Oh,
Yeah.
that's a sea cucumber.
Yes, thought it was a cuke.
This looks like a feeding trace of an echiurine worm, though, this sort of sun-shaped pattern here. They dig a burrow in the center and stick out their tongue like proboscis, but that's what that is.
Definitely. All right, making our way to the bottom. There's a little hermit or something running. He's fast.
And making our
sample tube.
Sample 2 of water for environmental DNA.
Did they take it?
Yes.
Okay. 2W, sample 2W on the bottom. All right, it's fairly barren. Well, barren, really. I only saw, 2 animals so far? Not including the infauna, which I can't see, but there's actually evidence of little lumps and mounds and holes. maybe some Thalassia detritus, which is seagrass. Not surprising since we're right off the Keys. Looks like maybe a spine.
Stream 3 has been changed to the multi-view.
Alright, we've got the multi-beam up on stream 3. Oh, sorry. The quad screen, Roland? Yeah.
Yes.
So these lack of ripples seems to would indicate not a whole lot of current seen right here on the bottom.
Yeah, I'm kind of surprised. I thought it would be a lot rip tearing through here. These were pretty deep. The surface currents, I'm sure, are strong. It really doesn't get super strong until that current passes between the Bahamas and Florida. So you decrease the space it can go, which increases the speed of the current. So out here, the current has a lot of space, so it doesn't have to speed up as much. But once you get around the Bahamas, it is like tearing through there. Right? The Florida current comes ripping through there, which just causes a lot of problems for us, diving up there. Got some mounds at the top.
I got Santiago Herrera in the chat today.
Oh, nice.
It's great. We got lots of octocoral people. I hope they're all ready to call in and
Correct me.
help us out.
What?
Thank you. We'll be hollering.
Yeah, there might be a fish there. Oh, okay. Let's zoom. Can we zoom in at center screen? Yeah, that long one. Yep, it's 6. That guy. This looks like a small fish.
Ooh, wow. Have not seen this fish. It's got a head shield.
Yeah, I wonder if those are bioluminescent. Anyone know the name of this guy? Do you know the name, Emily? Okay.
See the more of those pteropod shells on the Sandy bottom.
Mm-hmm.
So I guess you weren't seeing in the chat room. That's
Oh,
the genus
Ipnops.
Ipnops, okay.
A weird...
Related to the codfish. It's in the same family, or related family.
Oh, it's related to the tripod fish. That's neat.
Its photoreceptors are spread over the dorsal part of the head with reduced eyes. Very, very interesting.
I just want to point out that our video clipper, Emily, knew what that was from like 45 feet away.
Yep.
Okay? So, yeah. She deserves credit.
Experience.
And she was pretty darn close on the name guess.
Let's get another good close-up on those eyes if we can.
You deserve credit. That's pretty cool. So these, like, some yellow plates are spread over the dorsal head, so they're really a photoreceptor. That's neat.
Excellent. I think we can move on, though,
There's another
now,
one
right?
there. Oh, yeah, we're good. We're good whenever you guys are.
Oh, yeah, there's another one, top right. Ipnops.
Ipnops. That's a good name.
Yep. That's the name that sticks in the head, at least for a few minutes.
Yeah. We'll see one again in 3 hours, and I won't have any idea what the name is. It's okay.
Collective knowledge of the community will. So it looks like we must be approaching this ridge from the west.
Oh, there we go. There's cucumber.
Yeah.
Let me get a zoom on that purple guy.
Holothuroidea. This looks like the one that's capable of swimming, I believe. Maybe.
Oh, like a Nypniastes? I don't.
I'm not sure. No?
Nypniastes is really red. Has this like collar around its neck.
I see legs under there. This is a scale worm.
You think so? Oh my goodness.
Well, maybe I'm
I
not
don't
right.
see segmenting.
No.
That long gut.
No, I think you're right. This is a
Flip 'em
cucumber.
over.
Cucumber, and I'm just being those ridges, those lines on this side had me thrown for a second. I thought those were little polychaete legs, but those are definitely on the cucumber.
The texture of the skin
They
to give
sure
it.
did look like parapodia as we were moving in, didn't they?
To my unpracticed eye, yes.
That skin is typical, right? That's down here in the real deep. These cucumbers usually have that weird. See the skin on the top of its head?
Mm-hmm.
Okay, its head. Sorry, if anyone is not sure what we're looking at. The top, the head of the cucumber is to the right, and the back end is to the left. So if you look at the texture, it's like this ripply feeling almost has to it. That's a lot of these down here have that same kind of feeling. I of course can't give them names, but
That's all right. It's not the Epniastes guys, according to Mike
Nypniastes,
Becchione.
that or the headless chicken fish, which I'm sorry is the best common name ever.
All right. Thanks for that zoom.
A little shrimp, maybe?
You see some rocks in the serious view.
Yep, some rocks ahead.
Excellent. Like a small sponge?
Yeah, it does, doesn't it? What's it doing out here?
And there's another one towards the left.
Can we get a snap zoom on that white ball?
Wonder if this is a small
Is it growing on a rock?
hyalinamid, maybe?
Oh, it has its own stalk.
Maybe.
That would make sense.
If it has a stalk, it's very short.
I've always wondered about these stalk sponges, what they look like when they're small. I mean, do they grow a long stalk and then the quote-unquote body grows around that? Or do they grow kind of like this and then the stalk elongates? It's an interesting question. We need a growth series on these deep-sea stalk sponges.
Mm-hmm. Well, do we want this one while we're here?
If you do. Nah. Yeah, you can see the pteropod shells, little white triangular shaped shells on the bottom. Here's an interesting poo line. It's different. It's got to be. Look
there
at
we
the
go.
oscule on
Definitely, yeah. Now that we got a little sharper zoom in there. So that's likely the family Hyalinamidae. Group of glass sponges. Called glass sponges because of their their spicules are made out of silica.
And they're so fine they could be, in theory, used for fiber optics, but that's expensive.
Yeah, although many of them do transfer light. Some of the long, skinny ones, actually down in Antarctica. There are some sponges that they actually have diatoms that live inside them that live off the light that's concentrated by the spicules that go through them.
Alan.
Yes.
That's crazy. Can we see this pink thing? I had no. That's how deep are those sponges at?
They can be pretty shallow in Antarctica, yeah. Because glass sponges are generally, restricted to cold waters. That's why we see them in the deep. But obviously, it's very cold at the surface in Antarctica.
So their depth restrictions aren't as shallow.
No.
Yeah. I don't know what this pink thing is. We have a pink blob.
You see another one of the holothurians up above it. Cucumber.
I'm sure someone will chime in with a name for that guy at some point.
I suspect the pink one is a different species of sea cucumber.
Oh.
I don't have a clue what that is.
Isn't that a good feeling?
Yeah, sure. Why not?
Maybe another
It looks like
cucumber,
a
says
tongue.
Scott?
It looks like someone's severed tongue.
Yeah, I think it's another sea cucumber with the anterior end raised off the substrate.
Okay, a cucumber.
Oh, yeah, and you can see its respiratory, anal, respiratory tentacles right there, sort of tucked in maybe. Okay, that's cool.
It looks like a tongue.
Has his head buried in the sand.
Yeah.
Different holothurian.
There we go. We got some solitary cup corals.
So our first look at something living on a rock on this ridge.
Right, you can zoom in on those. They're interesting. Look at the body form is really like almost like a V.
Mm-hmm.
A V shape. That's cool.
That's beautiful, and you can see the
tentacles and the correlate.
internal skeleton.
Mm-hmm. So there's well over 500 species of cup coral, and they are impossible to ID.
Don't tell that to Steve Cairns.
I know. Well, with a sample in hand, he can do it.
Yeah.
I don't know if he can do it from a picture. Maybe some of them.
Yeah, I would imagine some, but you're right. Many not. And that's
looks
different.
Like a totally different
different species. Solitary coral.
Cool. That one's really interesting. Looks like a daisy almost, or like a sunflower inside.
And that skeleton, so we're used to thinking of calcium carbonate skeletons of corals as really hard and thick and dense, and some of the deep sea ones are very, very thin and fine. So you pick them up in the back in the museum, and they're very light.
So there's another one of those cucumbers. They're the most common species out here. We've seen 4 or 5 of them, at least from what we can visually see without zooming in on all the sand into the tiny stuff. It's a pretty steep slope, though, 45 degrees maybe. Sediment veneer, sediment, there's thick sediment on top of it. The rock ledge here.
Scott Francis has been looking at some of his old pictures and recognized that both of these cucumbers probably come from the same family, Synallactidae. So we're narrowing things down. Find more rocks.
I know. There's some bioturbation coming up on the upper left.
It basically means mixing of the sediment by organisms.
Yeah, you can see the little pock marks and little mounds and stuff, so they're caused by infauna. Here's a nice circle.
Yeah.
Right in front of us.
A lot of those things make it into the fossil record, and there's a very rich taxonomy of the different forms of traces that animals' behavior leaves in the rocks.
Yeah, I was watching that on Eons too, on PBS Eons. That's such a great show. I highly recommend. And it's public broadcasting, so I'm not like favoring anybody. It's government funded.
It's
Watch it. It's awesome. That's where I learned about how the echinoderms were originally bilaterally symmetrical. Thank you.
Yeah.
And then they eventually became radial. So there's one species in the fossil record where you can see it. The mouth is closer to the top of the versus in the center.
I think they retain that bilateral symmetry in their larvae still.
We got a small rock here. Can we have the lasers on for a sec? Yeah, so it's about a meter wide. So the lasers are 10 centimeters for scale. So distance between the two lasers is always 10 centimeters, no matter how far away we are. So
Another cup coral there on that rock. Looks like that one with a longer, taller theca. Taller coralite.
Yeah. Yeah, it looks similar to the one we saw earlier. There's a couple little shells on here, maybe some snails. Looks like some encrusting sponges. That's what this fuzzy looking stuff is.
Yeah,
oh, there's a neat sponge up top with a really fine oscule. That's neat.
Yeah, that's one thing that's been a constant, well, at this depth and everywhere we've been, is if you look in on the hard substrates, encrusting demo sponges are, small ones are ubiquitous, many sorts. You can see just down in the bottom left something completely different than what's above the cup coral.
Right. We got a little Zoanthids too. It's at 9, 8 o'clock-ish under that rock.
Tucked under there, yeah.
You can see the little stinging cells on the coral polyp, the little lumps.
A little, yeah. A lot of cnidarians, they concentrate their stinging cells into little warts.
Oh, we're good whenever you guys are. We're looking for that rock wall. That's what we're looking for. And there's some more sargassum here that's floated down, that detrital sargassum.
On the right?
Yeah, on the right. It looks like maybe worms or something, and maybe hydroids.
Oh, yeah, I think there's a rich hydroid community growing on the top of that rock. You can see lots of very small colonies of hydroids. All of those upright pieces are probably...
Oh, that might be a black coral.
Oh, I think that's different.
Tiny black coral.
With really, really delicate... Now, is that something that's commonly seen? I don't know how common.
It's very tiny. I probably have never seen anything that small. It's just because D2's cameras are awesome. Go ahead, Scott.
I would agree. It's a black coral, and it looks like maybe it's starting to put up branches like it would be a Bathypathes. And so it's a relatively recent recruit. I mean, these things take decades to grow, so I don't know if I'm talking about 2 years or 20 years with that specimen.
Thank you.
They are definitely not uncommon at this depth.
Good to know.
That's an interesting mound.
So I noticed that small black coral had very distinctive white mouths.
Like in the center of the tentacle?
Yeah, in the center of between the central pair.
Yeah. Maybe a burrowing anemone. Oh, this is neat. Can we kind of just pan down and look at this crazy lump?
As you're zooming in, Alan, I'll say that like a lot of hydroids, some of these black coral polyps have like a raised hypostome. That's probably the white you were seeing.
Uh-huh.
What's a hypostome, Scott?
Basically, the center of the polyp, the mouth is raised on a little cone. Think of it like the lips of the mouth, if you want.
Okay, thank you. That looks like a Sarianthid, which is a burrowing anemone. They actually live inside a tube.
Yeah, they secrete mucus, and they use a very specialized kind of nida to entrain sediments to help make their tube, and their tubes are really interesting, very sort of rubbery, have an amazing consistency. So if we were to get near this, it would shoot back down inside its sock-like tube and disappear from view. And you can really recognize these guys because they have the smaller oral tentacles in the center and then the larger ones on the outside.
But he did not make that mound likely. It looks like something around this has made, there might be a little home in there, one connected home. I'm not sure what made that. Some more of these purple cucumbers. I wonder if these curly cucumber, if these curly poos are from the cucumbers.
Maybe.
Yeah. Because they basically, they're deposit feeders, right? They just put all the mud they're walking across into their mouths and pass it through their digestive system and pull out any living or bacteria or whatever their bodies can digest. And then the sand comes out. Got a couple small rocks coming ahead. The slope's pretty steep. Yeah, we've come up about 10 meters so far. I think this black spot is just rocks. No offense, geologists. I think this black spot is a very interesting rock. No better. I try, guys. Is that the ferromanganese, the black crust on the rocks? Isn't that what it is? Maybe.
I would guess, based on what we saw before over on the Blake Plateau, but
We're doing weekly drills, so no emergency on for real. So if you're landside and heard that ringing, that's a weekly drill. No need to worry.
we are safe.
Yes.
Just making our way up slope.
Mm-hmm. Yeah, looks like we've got some rocks ahead here. Nope. Yep, a couple rock outcrops. So a couple meters of rocks exposed here, but mostly sand. So we go from 100% sand to maybe 80% sand with some 20% exposed rock. We're waiting on the ROV to settle out so we can get closer. From a distance, the rocks are pretty barren. I would imagine if we zoomed in, we could see a couple of little things. Maybe a sponge at the bottom of this one. There we go. Little white ball sponge there. Maybe some hydroids growing on it. Yep, some tiny little sponges, encrusting sponges grow. There's a dead cup coral.
On the Explorer, we have full muster. We have full muster. Electrical isolation complete. Electrical isolation complete.
There's another dead one over a dead cup coral on the right-hand side of that rock. And maybe some dead Faria at 9. That's
yeah,
what
although the its folds are very wide. So
maybe some hydroids here. Whoop. Something in the water. Oh, maybe that's Bathopathes again. A very tiny one.
yeah, I've got to think the
Get the polyp look. Yeah,
This
definitely something.
polyps so tiny.
This one looks like it's been through some hard days rather than being a young one. It looks like a lot of broken branches. At first I thought the branches were subalternate instead of being directly across from one another. They were alternating, and that's one of the characteristics you can use to identify some species. But a closer look there showed that they were broken up. So maybe something that's old and had some other material tumble down on it and breaking off some of the branches. I don't know what would feed on the branches.
Pretty beat up.
See a bunch more encrusting sponges on this rock.
Looks like a dead cup coral, and then on the top of it is a barnacle, a little gooseneck.
Yeah.
Explorer, ventilation has been secured. Ventilation has been secured.
That's interesting. Is it Lapos? Is that what they're? I think so. The gooseneck barnacle. Another Bathopathes at noon. That one looks beat up too.
Mm-hmm.
Yeah.
There's a couple of them there.
And that brown here, that's Thalassia. That's a type of seagrass that's floated down from the surface or shallower depths.
Yeah, so this Antipatherian It looks a little ragged, but it's been pretty common. I've seen a number of colonies of it.
Another cup coral.
There's the other.
Maybe some tube worms in the background.
I was just thinking that this black coral has tissue that's thin enough and white or transparent enough that you can actually see that the skeleton is black. Very often you can't see that in these black corals, and you see instead other colors, especially orange, yellow.
Yeah, the orange tissue on top of it.
There we see some larger rocks.
It's remarkable on that black coral, and actually a lot of black corals, how thin the tissue is, how little tissue there is relative to skeleton. You can see that thing there. It's pretty hard to discern the soft tissue polyps.
Yes. Got these black encrusted rocks. There's something center screen. I wonder what that is. This brown.
Yeah.
Maybe a sponge or something.
Donut-shaped sponge, too. Donuts.
Yeah, that's interesting.
Sure, that is a sponge.
Looks like the sponge next to it in the center, but then like the hairy stuff caught.
Yeah.
I'm not sure what's going on there. I think it might be just based on the center of it. You got that like white tissue in there. It looks spongy.
Just has all these spikes around the outside. Interesting.
Yeah, it does look like a sponge that's just accumulated a lot
tan
of
at
particulates
11
and
o'clock.
sediments around here. And I know typically, when I hear Chris Kelly describe them, he's referring to that part of the sponge tissue being dead, but it certainly looked alive on the inside. At the center of the donut there.
I don't even know what to do with this. What's going on here?
It's like it's been
So
bored out.
Yeah.
I wonder if a boring sponge.
Yeah.
Boring, not as in not interesting, but a sponge that has some of the sponges they etch out through the rocks or the other substrate, and they live beneath it, and then they stick their oscules. They make little holes, but I'm not sure that's necessarily what's going on here, though.
Yeah, this is a weird one. I guess that's a serpulid worm, a calcareous tube that you see running on top of it.
I don't know.
Yeah, I think so.
Yeah.
Yeah, polychaete worm tube.
Oh, there's a squat lobster at 4, just below the bacterial matting. It's weird. I don't know what I'm looking at. I don't know what to call that.
I wonder also if could any of this be really old reefal material that's been coated in ferromanganese crust, and we're seeing some erosion there in the underlying, I don't know, coral holes. I'm not sure what it is either.
Could be.
It's a hypothesis.
It could be worm tubes, too. We're looking down into worm tubes. Yeah, there's a little, might be a clatorizid, maybe. A little sponge. A little circular.
Was that
Well,
there?
it almost looks like it has a pointed back to the thorax.
You guys see the brittle stars hanging on a very small
The brittle stars?
something
In
that
the
is below
foreground?
the squat lobster?
In the foreground below, yes, below the squat lobster. Thank you, Roland.
Not sure what it's growing on.
Looks like a hydroid or something. This is what I'm here. Really teeny tiny.
Or this sort of webby material that I suggested a few days ago maybe would be released by sponges that are releasing gametes. Still don't know. It's a hypothesis, but I think it's an interesting one. We see it not infrequently associated with rock ledges where there are sponges nearby.
I missed the first part of what he was saying. What was that?
Was there sort of a mucousy net on top of the sponges? Just sediment filled?
More having to deal
But
with
maybe
their reproduction than it being like a bacterial mat that I was calling it. Yeah. There's a yellow sponge on top of that rock, too.
So far, not seeing the large octocorals that we were maybe hoping to see along the edge of this ridge.
Not yet. We should be able to get up to the top of that nose, and it will probably increase in hard bottom. And the sediment will.
Yeah, I think
Yeah.
when we did this ridge stuff last time, they were really up on the ridge crest itself, and I don't think we've reached that yet.
No. We've only come up, let's see, we're at 230. We've only come up about 20 meters. This is an interesting little coral.
Looking bamboo corally.
Yeah.
Bamboo-ish.
No, I think.
What do you got?
Now I'm thinking it's a primnoid, actually. Yeah. Notice how the polyps, each of the polyps looks like it's kind of ridged or ringed. That's because of the plate-like sclerites that overlap going from the axis towards the tip of the polyp. And then the other clue is that the polyps are arising in these little clusters that we call whorls. And I can't quite tell if there were 2 or 3 there, but that's definitely the family Primnoidae.
Fire drill and report to the MESTEC for debrief. Report to the MESTEC for debrief.
Hey, sorry, Scott. They're doing fire drills, so it's hard for me to hear. I think they're done now, though.
Yeah.
Okay.
I think he was asking if there were 2 or 3 polyps in a cluster on that
Yeah.
on that primnoid.
It looked like 3 to me.
Yeah, I think so. And we don't need to go back and look. We had a good enough view.
Great.
Just noting that can be a variable. That is a variable that are in part species diagnostic. So when you're looking to identify something, that's one of the characteristics that you would look at. But no question, that was family Primnoidae.
Yeah, we're just looking at the quick replay. I think it's going to be good enough to figure it out.
Yeah. Yeah, we're going to make our way up the rest of this slope. I think there might be some...
The family Primnoidae is one of the most common in the deep, deep sea, that is below 1,000 meters, and very diverse, a lot of genera. I think there's more than 60 genera, and is another pet project of Steve Cairns, who's described a lot of species and genera in that family. Steve Cairns, former curator of Cnidaria at the Smithsonian Institution, National Museum of Natural History, who recently retired, but certainly hasn't slowed down in doing his work.
No, he hasn't. He's probably more free to do the work he loves.
Which is pretty amazing given how productive he was when he was a curator.
We got a cool rock view coming up. This looks like probably the habitat we are looking for.
Oh, we got a big jumble of rocks in the background there.
Maybe a bamboo coral at 6.
A little cup
Can
coral
we
there.
pan down a little? There you go, that little coral there at 6 o'clock. Yeah, it looks like a bamboo to me. They're not seeing the rings, though.
Yeah, good call on that one, Stephanie.
Okay.
I would agree with you.
We're getting a little worm up above on that
neat.
rock.
And some dead cup coral. Okay, we're good. If you could just pan up a little bit when you're ready.
I'll get a good look at this bamboo. It's hard to make out the nodes, but I think I can see them in there.
It looks like it's bent its center stalk.
It's unquestionably a bamboo, whether or not we see the nodes, the shape of those polyps. There's something interesting going on. What's down below? Is there a jelly on one of those polyps on the lower part of the colony?
Can you pan down when you guys get a chance? Yeah, what's going on
A
there?
little bit odd.
It almost looks like they've just pulled their tentacles in.
I do.
Oh, is that all I'm seeing?
I think so. Like they've rolled up.
Okay, maybe it's a really weird angle.
Yeah, it doesn't look like a jelly on them, like you guys
Yeah,
have
can
seen
we zoom
out
in on those weird
Pacific.
polyps anymore, or are we as tight as we can go?
It's interesting. Yeah, maybe
Max,
Stephanie, you're
okay.
right. I don't know that I've ever seen just the tentacles pulled back with the oral disc still open like that. It looks like a little trumpet or something from my angle.
That's cool.
You just never know what you're going to see down here.
Yeah, right.
Mm-hmm. We're going to get a little bit closer and tilt down and see what we see.
Yeah, I'm not sure we can still see it from this angle.
Oh. Only one way to
Yeah.
find out.
It'll obstruct it a little bit. But yeah, sort of.
well.
You can sort of see it. Ooh, hello.
Little amphipod.
cute.
Oh my gosh, that's great.
See if he gets eaten. That's probably a little too big for that coral. Yeah, we got obstruct. It's okay, guys. Thank you. I do like that little
Yeah,
amphipod.
good.
That
I
was
think
cute.
you're right, Steph. It's just those tentacles. I guess they're not so much withdrawn as just curled a bit, but the rest of the polyp wasn't contracted at all. So it's a little bit unusual. That's cool. Just on 2 polyps.
Yeah, it's
So
weird.
I wonder if they had just captured something and had brought it into the pharynx.
And haven't really contracted all the way.
You know, it's interesting. All of the many, many observations we've made of these deep-sea octocorals, it's very, very rare for us to actually see anything with some prey that it's captured and ingested. We do occasionally, when we collect them, see, you know, and looking inside the polyp, we might see something. But sometimes I wonder, you know, when do you guys ever feed?
Right.
All these tentacles hanging there, and I never see you catch anything.
It's the testament to how little the food is, to how little the little food is. Help me. Give me my sentence. You can't get it. How little food there is.
There's
Yes.
little food
Thank you.
available.
The low density of food
Thank you.
availability.
With which we see it is going to be lower.
Here's another one of those weird holothurians that looks like a tongue. The pink one.
Synallactidae.
We had a name for that guy?
Just the family name. Synall.
Oh, I thought that was the purple cucumber.
I think both are in the same family.
Oh, okay.
All right, so we're getting up to the bigger rock. See a little trail where sediment has fallen down that slope.
You want to get a water sample at the base of the wall? You would rather do it at the top. We got one at the bottom.
We got one at the bottom.
That was how long ago?
Let's see what we see.
450, yeah. 25 meters deeper we were when we landed. Yeah. Okay, it's pretty far away then.
So this is clearly a much larger chunk of rock.
Mm-hmm. I'd imagine this is what most of this, the base of this is.
Mm-hmm.
You know, probably see most of this the rest of the dive, based on the multi-beam. There's something
There we
up
got
there.
a colony up there. Yeah, 2 o'clock.
Yeah, it looked like really fine Gorgonian at 9. Did you see that?
Mm-hmm. It's like a little antenna wire.
Shape. Yeah, the shape and cluster of that fine branching suggests bramble-like bamboo coral.
Can we zoom in on that it's almost center screen right now? Right below center screen, it's about 6 o'clock. Actually, there's a few of them, so. Yeah.
Something in there too.
Kind of hard to see. There you go.
Yeah, things have a good way of blending in.
Mm-hmm. Really fine. Oh, that's different. It looks like a bamboo coral for sure. I've never seen one that looks like that.
Yeah.
Any ideas, Scott?
Weird, because it looks like the structure looks like a bamboo coral in terms of the colony and the branches and the way the branches are going out at all these odd angles. But the delicate polyps actually look like chrysogorgid polyps. I'm sure they're not. This is going to be a bamboo coral, but kind of interesting.
They're very fine.
Yeah.
The polyps, the tentacles on the polyps are much longer in relation to the actual polyp.
Mm-hmm.
Right, yeah, exactly. And note that the polyps are very evenly spaced. They're all on one side of the branches. There's the polyp body is really short and squat, and then those tentacles are hugely long. I don't recognize that, and so.
You want to collect it?
You want to collect it, Scott?
Yeah.
Can
Yeah,
we
that's what
can
I'm
we
trying to
collect
say.
this?
If we see if there's several up here, that's a collectible. I'm not sure if that's an S1 clade. I sort of don't think so, but the delicate nature of the branches suggests maybe it is.
Another amphipod just went swimming by.
So it's exceedingly difficult to collect on a wall. I believe they're going to try. That's what it
Sounds like. But this looks like an interesting collection. Certainly not anything we've encountered on our expedition so far.
I was so focused on the polyps that I wasn't really paying attention to the nodal, whether it was nodal branching or internodal branching.
I honestly didn't even see the rings.
Yeah. We've Les Watling and I have collected at least one bamboo coral, two different collections of it that we found in the Nashville seamounts that had no nodes whatsoever. It was definitely a bamboo coral, genetically and otherwise morphologically. But at first when we got it, we couldn't even put it in a family because we were so flummoxed by the lack of nodes. And it also had this kind of delicate branching. I'll have to look it up in the database. It didn't quite look like this, and I don't think the polyps were like this, but
It's really fine. It's very different.
And that
Look,
one
and
ended
even
up
like
in the jade
even the
blade.
branches seem abnormally long compared to the other
Yes, and
species.
that would
we've
be
seen.
a cladorisis characteristic, the S1 clade that I noted at first. And that genus cladorisis was named because the colonies are so delicate and they break easily.
Okay.
Yes. How much of this, Scott? Gonna-
Ideally, you would take a clipping below a point in the branch, so you had the branch point.
Okay.
That
So-
may be difficult with this thing, but that would be the preference. Otherwise, whatever is easiest for the pilot to get
Yeah.
under the conditions.
So as much as you guys can get, ideally.
Yeah, this is the problem with these things. The branches are so far back.
Yes, I mean, yeah, that's fine if you can do that. If you can go a little deeper, that would be, yeah, ideally that. Yeah. How about in between the base and that first branch, or after the first branch? And that way he can kind of grow back a little bit. I saw more than once.
Yeah.
Take the whole thing.
Mm-hmm. Yeah. So if you're just joining us, we are making a collection of an unusual bamboo coral colony. Okay.
Oh, Caryoptosis? Remember the family?
Keratoisididae.
Okay.
I think just stick with that.
It doesn't want to come up in worms. That's interesting.
Oh,
It's in the deep sea one, but
If you're talking about in C-tube,
yeah.
our version of C-tube has not been updated to the latest version of Word.
Okay.
nice,
So you will
nice
find
job.
Keratoisididae, you'll have to type in Isididae and then in the comments box put Keratoisididae.
Okay.
Paper just came out last month, and so
Thank you, Scott, for that clarification. It's a challenge getting this one in the box. Just large. Port enter. Yeah, it's mostly down in there. Yeah, it's got nodes for sure.
Thanks, guys.
Great work.
I'm trying to avoid putting anything in the outer box.
Challenging collect. Yeah, I can see that.
I think all of it went in that box looking from above.
Ah,
So that's good.
great. Thank you very much for that collection. It'd be interesting to see if we see that again.
Yeah, I thought there was at least 2 colonies, but maybe looking at it now, it was one. It was one that looked all beat up, was separated a lot. I think there's one right there at right off screen at 11-ish.
It does look sort of
Looks like
similar.
another one, yeah, one at noon too. They're really fine. Hard to see. Yeah, just snap zoom right there. That looks like another one.
Yep, think so.
It's really fine.
It's so interesting to me the way they grow in this very open pattern. It's almost like there's no sense to the direction that the branches are going from one column to the next. There's no clear pattern. And so it's not like a clear fan or a clear bottle brush or anything like that. Must be really responding to environmental conditions at the time that the branches are put down, I would imagine.
Yeah. There must be a lot of upwells and changes in water direction. Which I guess makes sense up on top of this, shark fin ridge that we're on. It could be easily done. We're good whenever you guys are ready to go.
Delicate branches. Amazing it doesn't completely get crushed. So I wonder, the fact that it's so delicate, I wonder if right now we're in the lee of the current and not the strongest part of the current, and if we were in stronger current, you wouldn't see these? Just another
possible.
hypothesis.
Maybe when we
so
get
many
on.
hypotheses to come back and test,
I know, right?
So
yeah, we're on this really stark wall, huh?
Yep, we've come up about 30 meters since the start.
There's another cup coral.
Yeah, looks to be that same species we've seen a lot of. Nice to see it doing well down here.
Maybe some worm tubes. I'm not sure what that is hanging off.
What's that dangling down? Yeah, let's take a quick snap at that.
Foothold of something, maybe?
It's up against the wall. Oh.
Oh, no, it's did Hexactinella or... That's odd.
Yeah.
Okay.
Thanks. Yeah, it might be I think it is actually the wall of a thin hexactinellid. What's the name I'm trying to think of? It's one that sometimes has these little side branches off of it. Maybe we'll see it again.
I'm pretty sure that one was dead, yeah?
Yeah.
Oh, we got Faria up here. Looks like partially sedimented
Really,
over.
yeah, really wide open flutes on that guy. The tubes.
Oh, Chrysogorgid at 9, looks like. We have a request from Mary Wigstein to get a Chrysogorgid with a Anamura in it.
Ah.
So she had an interest in... I'm not sure if she thought they were a new species or just undescribed.
Yeah, good memory, Stephanie. And she was telling me at the time when the line was disconnected from the ship that it's specifically the small pink ones, and she said they are undersampled and unknown in the Gulf of Mexico.
Nice. What about this guy? He is pink.
Yeah, this is. This qualifies. This is
Yeah.
really long arm.
He does have really long arms, doesn't he?
I wonder if Steve Oskevich is still in the chat.
The squat lobster.
The squat lobster, but if you have to get coral.
Yeah, I don't think he's not going to let go of that coral, so your best bet is to grab the whole coral and the squat will
Or
come with it.
just the part with him.
Yeah. Yeah, that's the part that he's above. I missed what Scott was saying at the end. I'm sorry, I had to talk to the pilots.
That's okay. I was wondering if Steve Uskevich was still in the chat room because I think he's been on some other cruises in the Gulf, and he may know more about which of these carcigorges with the crabs have been collected.
Not 100% on the crab. We're not 100% sure on that. We'll see more.
Yeah.
You want to keep going? Let's keep going. We're going to keep going. Sorry, guys. I didn't mean to cause a drama. Yeah, we're fine. We're going to wait on that. I'd like to get more confirmation on, because we're limited on space, so. Oh, that's interesting. Yeah. Benthopathes. We're looking straight down on it.
Yeah, we are.
It's hard to see, but its polyps are really poofy.
I'm picking up on
This one
that.
might be something called heteropathes.
Heteropathes.
I'm saying that because if I recall, the heteropathes near the main stalk seem to have this real dense collection of polyps, and that's what I'm seeing looking down on this. I'll look it up and make sure I'm giving you the right name.
Awesome.
That's really
that's
nice.
neat.
You can see that dense set of polyps right there in the central area. Real increase in the pigment too. That's what's
They're very fleshy.
heteropathes. Well, that's a new taxon
Yeah.
for our trip for sure.
We're good, guys, whenever you are. There's worms.
No.
No?
No. That's an octocoral.
Oh, nice.
That's another one of those cornularids or something like.
The ones that live in the tubes.
Yes, the ones that live in the tubes. Stoloniferous, maybe.
Well
And
done, Alan. Yeah, for sure. That's great. Great observation.
Another one of those bramble bamboo corals that we collected.
yeah. I think a Niskin about now would be a good choice. Thank you.
How many do you have, Alan?
This will be number 3.
This will be number 3.
I'm sorry, I meant how many do you have available to use?
5.
5 total.
So you'll still have one more that you can take near the bottom, and then another one as you're going up.
We took one at 500. We took one when we hit the bottom.
Yeah, I'm talking about going forward. I'm just thinking we're going to get to a place where there's going to be a lot more coral, so I just want to be sure that you still have another one to shoot at that point.
Yeah, we should.
Yeah,
One in the center,
we'll have
and
2
then one at the
more.
end. Yeah.
Very good. Okay.
Looks like a yellow gorgonian at 9.
Yeah, that looks like a plexaurid from here. Something like what we saw yesterday, the Paramurcia or any Canthagorgid maybe. I like that the coral diversity is picking up here as we're going upward.
Right?
Yeah, as expected, right?
You think Paramurcia?
Maybe. It's kind of a scrawny looking one. I don't know. Yeah, it must just have all the polyps withdrawn.
Paramurcia, you think?
Yeah, I mean, that's what I'm that's my go-to for these sort of yellow Gorgonian sea fans.
Mm-hmm.
It's almost certainly in the Plexauridae. Yeah, I see Santiago Herrera is agreeing, Paramyrcia. It's just got all the polyps retracted, so it looks a little odd.
There's a small colony on the wall behind it.
Oh, yeah. What do you think that is?
I don't know, that looks something like that we sort of also saw in the background of something else that I was hoping we could get a look at.
It looks bryozoan-ish.
It does look like a bright bryozoan. It's white. It's at 10.
Yeah, in the background. There you go. Yeah, that's what it is.
Yeah.
Okay, thank you.
Thank you. So third water sample is done. And there we have this. I'm pretty sure it's a bryozoan, right?
Mm-hmm. I think so. I'm voting for bryozoan.
Classic.
Look at this 90-degree wall.
Look at this
Nice.
wall. All right.
Got a solitary stalk, probably bamboo. Pretty long, it's 1.5 meters maybe. Another Paramyrcia, yellow, at noon.
Yeah, so this is great. This is what I was hoping we would start to see, and it is, reflective of what we would see in an offshore seamount. I think it has to do both with the depth and the fact that we have these exposed rocks here.
Mm-hmm. There you go. You can see the rings. They're pretty far apart.
Yeah, good, Stephanie. Very long internodes. The internode is the name we use for the white part of the skeleton, and the node is the part we use for the brown part, what you were calling the rings. Those are mostly protein, whereas the white part is mostly calcium carbonate. This is something I think that Les Watling and his team would call long bones.
Long bones.
And they grow as these unbranched whips. So
Yeah.
the genus they are working on, they're getting pretty close to being able to publish a name for this, but characterized by these very, very long internodes.
Really neat. See the hold fast.
Really nice.
You can also see the diameter of the skeleton there is pretty thick towards the base. This is a pretty old one. You noted, it's pretty tall, more than 1 meter tall.
Yeah, he's
So it's probably been growing here for quite some time.
So Santiago is asking about the possibility of collecting that yellow Paramyrcia if we get them again.
Isn't there one right there at noon?
Yeah.
Is that collectible, guys?
Would be especially nice if it had the ophiuroid.
Okay. Let's definitely get one with an
isolate.
ophiuroid then. I think if this one doesn't, we could find another.
We could find another.
Yeah.
Maybe we take a quick zoom and see if there's an ophiuroid in there. Doesn't look like it.
No, it's pretty though.
Yeah, so let's not make this a target for collection and hope. Oh, it's nice. This one's
More sturdy looking than the last.
and the polyps are out, so you
Yeah.
can.
Yeah, that's one of the differences. This one has the polyps extended. I do want to make a note that there has been some work on how close an association there is between the ophiuroids, the snake stars, and particular corals, including in the Plexaurid and the Paramyrcia group. And one of the early suggestions, I've noted now we've seen 2 Paramyrcia that don't have an ophiuroid, a snake star.
Right.
And that's a little bit unusual for that genus, and it may be an indication that this is actually a different species that doesn't host the ophiuroid. So we do want to consider that as we go forward.
Would you suggest that we get this while we see it?
Well, I just made the comment before that when you say, well, we'll wait till later,
Yeah.
may not find it later, and that's partly because things are so depth stratified in the deep sea. And especially in this area between 2,200 meters and 2,000 meters, there's a transition. So I get worried when we say that. So I'd like to hear, if Santiago's still there, what does he think? If this is Biscaya on the seamounts, I'm pretty sure has ophiuroids, but both the ones we've seen here don't, and I don't really recognize the shape of the branch pattern here.
Oh, he's saying yes. Yes, please.
Yes, please.
Yeah, can we collect this?
Yeah. And this is one, you could see it's kind of waving quite a lot. It's got a softer proteinaceous skeleton. So this is one where a clipping of the branch with the scissors part of the claw is going to be required.
Just a clipping?
Which snap. They don't snap easy, but they cut easy.
This is going to be number 5B.
Yep.
Paramuricea.
Paramuricea, and there's maybe Biscayi. I'm going to check how confident that actually, maybe we should, just as a field ID, keep it at the genus, and then it can be narrowed down. Shrimp going by. So we're just setting up for a collection here. Just some context for the importance of this collection from Santiago Herrera in the chat. This looks like a species that's known in the Gulf of Mexico, but this is kind of far south from where they've been collected before, and so it could be a different species, or also this could be a potential location for a source of larvae. for populations that were impacted by the Deepwater Horizon oil spill farther out in the Gulf to the west. So. Only if you're concerned about losing port inner, but otherwise it's fine. I trust you on that. No, that is not a concern. A clip would be sufficient, yep. Yep. Yeah, that would work perfectly. Nice. Yeah, I think I'm going to step away and I'm back in a moment.
Yeah. That's awesome. Thank you. Are you aiming for outer or inner? Inner? Outer. Okay, thank you. This is starboard inner. Thank you. So if you're just joining us, we have just collected the second Gorgonian today. The first one was a bamboo coral, and the second one was a Paramuricea. That was the yellow one we just picked up. Yeah, sure, if you can get one without causing an avalanche. It looks kind of scary right here. Do you think those are loose? Yeah, if you can safely get one without causing a drama, then I'm totally down. So it's really important that we do collect these deep sea corals. Their genetics and their the ability for us to expand our knowledge of science and the species that are out here. A lot of these species have never been collected before. A lot of them are undescribed, new to science completely. There's also some cool video interactions we can get. For instance, we can see like the Paramuricids and their close associations with their ophiuroid symbionts. A lot of those are undescribed and understudied. It's really difficult, and just until recently, with the invention of modern-day technology like the D2 or other deep submergence vehicles, we haven't really been able to get a lot of these species. Most of the data that we have, pre-modern era, is from trawling. So a lot of the species we've been able to pick up, the past couple 150 years or so, have all been trawling data. And the act of trawling can be really detrimental to these individuals when they come up in the nets. So a lot of the times we don't get whole pieces, or we get little destroyed little chunks. But when we come down here with these really precise ability and fine tuning of these ROVs like D2, and our pilot's ability to gently reach out and snip little pieces of coral exactly the way they're growing in situ is really remarkable. On top of the fact that we can get the footage before we actually pick them up, so we can see what they look like in situ or on the bottom in the water before they're actually collected. It also is good for genetic connectivity. We do there's a lot of genetic studies going on, and every piece of coral that we can get a bit of DNA sample out of, we will do. So every specimen we pick up, we grab a tiny little piece that goes into 95% ethanol, and we're very careful not to cross-contaminate. We wear gloves, and we keep the genetics separated. So that's why each coral or each animal we pick up goes in its own box to keep cross-contamination from genetics from happening. And those genetics can actually help determine where the species break phylogenetically. Sometimes we can actually rename or put these corals or other fauna into different families or classes of species. So it's really just expanding our knowledge of deep sea fauna. And what actually lives here. Yeah. Sure. So we're going to collect a rock here, hopefully. Oh, it is loose. That's good. Go for it. We also like to collect rocks for geologists. Rocks can tell you a lot about what the underlying substrate's made of, how old it is, how long this feature's been here. I honestly don't know what else they look at. For the ferromanganese crust, I know is indicative of how long this rock has been exposed to open water. So that's that black coating along the outside of the rock. Here the rocks are probably very hard, but where we were the other day, the rocks were really soft and loose, almost like a clay, like it had compacted the sediments enough to make a clay, but not enough to make a sedimentary rock. So that was really interesting. It's different, just a totally different habitat here than there was in the dive yesterday, where we were off the Tortugas, I'm blanking. Valley, sorry. So that the Tortugas Valley, we noticed too. We were expecting, when we got down on the bottom, we were expecting there to be a tall mountain or ridge, and it just wasn't there. And that's because of how volatile likely the bottom is and how loose the sediment is there compared to like a place like this where these rocks are not going anywhere anytime fast, maybe rolling down the hill a little bit, but for the most part, this ridge is probably a really solid, non-moving object. That's really square. So it looks like the bottom of that is white, so that probably hasn't been exposed to the water. That's interesting. That's cool. Looks almost like a rust color. So that's going to be sample 6G. Port forward. I did it on my side.
Right. Bye. Ooh. No, let's take a look at that. That's neat. Yes. I think that's a glass sponge, unusual looking. It reminds me of Farreid. Yeah, this would be an unusual form for Pharreid to grow in, but there's something about the way it was, the folds on its body. Oh yeah, that's really nice. I think mm. We're going to check if that is feasible to collect that sponge or not. If this is feasible, it would be a really nice sample. Yep, dead center. Fantastic. Mm-hmm. Mm-hmm. Yeah, oh, for sure that's okay. I think that. So we're moving in for a collection attempt at this glass sponge in the family Phoreidae, or likely in the family Phoreidae. We are about 2,408 meters. Pilot, it's likely to be fairly brittle. Yep, that's all we can ask. So much smaller than I expected. So well done. Mm-hmm. It's a twofer. So it looks like We might get a bonus collection here. We'll see. I think we've already collected that. Yep, I understand. In the meantime, I get a great view of that wall. Yep, I see it. Maybe. Wow. If they mix, it'll be okay. Yeah, starboard inner. Yep, that's going to be fine. Better than losing it, for sure. Yeah. Oh, of course, of course. Yeah. Whatever's, yeah, most feasible. Yes. Nice. Good catch. Okay. And it went into, is that canister one? Thank you. Great job. I was holding my breath. I guess we can start making our way back up this slope. Really nice stark wall. I think it would behoove us to move up. No. Yes, we got a beautiful farreid. I didn't mark anything in there. I was distracted. That's when it was ended, and it took about 10 minutes. It was a very delicate operation. Mm-hmm. Family.
Lucidity? FA.
Yeah. All right. Great job. I think we got a little bamboo in the sample as well, because it was sort of hung on.
Here's 7B.
Just look at this wall.
Oh, it's crazy.
So in the interest of time, since we didn't, took a little while to collect that, just sort of urged our way up. So we're above 2,400 meters now.
Oh, nice.
So
The top of this is at 23, so about 100 meters deep, shallower roughly to go. This is exactly what I was imagining it looking like from the multi-beam.
Mm-hmm.
So this ridge, pointed ridge.
Ah.
These rocks are really rugged. Ooh, there's a cool gorgonian. Yeah, sure. Yeah, snap zoom on that. Look at this wall. It's like almost 90 degrees, 80 degrees. It has the same, it has really zigzaggy pattern. Not even sure what to put this in yet. That's beautiful. It looks like a stylaster, yeah.
Oh, it's just massive, though.
It is. It's really big. And it's got the little tiny lapos barnacles on it. You can see the little tiny round. That's really nice.
I wonder how long that took to grow.
Oh, geez.
A long time.
I don't even know what their growth rate is. Usually this stuff in the deep sea is very slow.
Has an aplicopheran on it somewhere, Scott pointed out. That's a small worm-shaped mollusk.
There's a mollusk at 11.
Yeah, I see the gastropod. They're usually wrapped up on a branch. It's possible. Could we zoom back in?
Maybe here?
Conference call's been reconnected.
All right, great. I'm back. Or you're back.
Yeah.
Yeah, I think there are 2, right? Or 3. 4.
Yeah.
I think there's a bunch of
Well, that would explain why there's so little tissue on this coral.
Huh. Thank you.
Yeah, I'm seeing at least 4.
Mm-hmm.
These are all the curled up brown.
Yeah, like the little tubes.
So they're feeding on the stylastarin?
Yeah. The apocopherans are specialized, as you said, Alan, worm-like mollusks that specialize on feeding on cnidarians.
Yeah.
Selenogasters is another term. This might be coralliid.
A coralliid?
Corals that has no tissue left.
Yeah, I think I saw one larger polyp sticking out from the other side, and it was confusing me because that was not what I would expected from a stylasterid. So that makes some sense. So that would be the common name precious corals.
Mm-hmm. Again, we're on this really steep ridge. It's quite thin. We haven't reached the top yet. Top should be another 100 meters. We got something at noon hanging off that. Really ragged rocks. They're not really rugose. There's not a lot of hidey holes. There's not many fish. But they are really jagged piles. It looks like a dead stalk of a coral. Maybe a small sponge or something growing on it.
Mm-hmm. I think
These little
that's
polyp
exactly
things.
what it looks.
It's these little polypy things? I wonder what those are.
It looks like
Yeah.
dead coral, right?
It's zoanthids, I think.
Oh, they're alive, yeah, and they are. I think they are zoantharians or zoanthidians, depending on who you are. Potato.
Yeah.
Translucent coral on the right of the rock.
Yeah, there's a gorgonian. Looks like stem of a gorgonian. Might be dead.
I don't know if you noted the small crinoid.
It's a crinoid.
But I agree, yeah, those are zoantharians.
Looks like we got the top of this ridge here. Just flattens out at the top. Where is the sand coming from? It's blowing down from the upper wall, maybe?
Great view on Stream 2 from Sirius.
Wow.
Yeah, that is a wall.
Stream 2, it does have a great view.
See some corals hanging off the walls.
Yeah, can you look just below camera? I think I saw something fun. There's like a white, there's white stuff.
Oh, yeah.
At 6? Yeah. It may be nothing.
Cat 2 view is great to see D2 in context of the habitat that we're in, and it's also showing how much Sirius is moving as it's translating the movement from the ship.
Oh, yeah.
Make us all better appreciate this whole dual body system of the two ROVs working together and the really stable images that we get from D2. Very nice.
Yeah, D2 acts like a really sophisticated clunk weight. I'm sure they don't want me calling it a clunk weight, but that's ultimately its function besides, being on
9,000
top.
pound clunk weight.
Yeah. Oh, okay, it's just a white rock. Was it fresh break off?
Yeah, sheared
Okay,
off.
we're good. There's actually some more corals on the side that are sticking out we can zoom in on. I just wasn't sure what was going on there. Super light area. Yeah.
Yep.
We can make our way over that way. Oh, yeah. Okay. Yeah, sure. Let's see if we can get anything ID'd from this far away. Yeah, that's tough. But there's some gorgonians out here. Maybe some more stylaster.
Seems like that's the side of the wall that is seeing the current. Might be more conducive to
Life.
Large colony growth anyway. I think there's life on both sides.
Yeah, that's fine.
Great.
Yes, we should be on this first nose of this shark fin ridge.
Yeah, we can just keep riding it up.
Yep. Very sedimented on top. It's very thin. A couple meters wide. Yeah, that'd be good. So I can barely see the lasers. Oh, what is that?
Something 5 o'clock. No.
It's probably a dead stalk of something. There's also some yellow sponges at 9. So we've got this weird wiggly line here at like 7. If we could zoom in on that real quick. There you go. It's dead center now. Yeah, that looks like the stem of a really thick stem of a Stichopathes or some kind of old coral.
Okay.
No branching on it.
Coral. It's coral rubble then. Only it's isolated.
Yeah.
So it's not rubble.
You get one coral rublé. I don't know what that is.
The way
Rublet.
it's twisting like that, I would venture to say that that's an iridogorgia. It would be nice if that's signaling that maybe we'll see an iridogorgia up ahead. I don't think we've seen any on the expedition so far. Alternatively, it could be a bamboo coral whip that's just highly encrusted with ferromanganese.
Looks like it broke off from right there.
Yeah, it does look like it broke off right there.
Another crinoid on it. Some maybe hydroids or bryozoans in the back. What do you think, Alan? Are those hydroids? What are those?
I would guess hydroids, but I'm not 100% sure from...
Maybe they're dead? Really short?
Well, they don't look alive, but sometimes they're just all pulled in. But it could be bryozoans as well. Maybe Megan McCullough in the chat can chime in what her thoughts on that. She'd say they're bryozoans. I'd go with that. There was something about them that didn't quite feel hydroid-like.
Mm-hmm. It's a lot of sediment accumulation up here.
Yeah, on the flat top.
Probably not too much current. Mm-hmm. Yeah, there's no rippling, and and I'm not really seeing the water move too much any one direction. Do you know which direction it's coming from? Both? Okay.
Mm-hmm. Hello, did someone join in?
Hello. Yes, I did. I was trying to figure out how to work this teleconference. Hi, I'm Danny Weissman. I'm calling in from the Deep Sea Coral Program at NOAA.
Oh, hey, Danny, how are you?
And how's it going?
Really good. It's a beautiful dive today.
You guys are finding so much cool stuff. And I know my boss Tom called in earlier this morning, and I wanted to talk to our listeners and our viewers a little bit about just some of the management in the Gulf of Mexico, some of the cool areas that are protected for deep sea corals.
Mm-hmm.
Yeah, go for it.
Yeah, so thanks everyone for watching today, and I just wanted to drop a few notes. So these expeditions are really important because we really can't protect what we don't know is there. So every dive and every expedition that we go on reveals us more and more about these really valuable underwater communities, as you're seeing today. And just to sort of talk about what we do at NOAA in the Deep Sea Coral Program. So we take data gathered from all these dives, and we store it in our huge database. And we have over 800,000 observations that include coral and sponges seen in the U.S., but also in international waters and in the high seas. And I was just doing a little number crunching, and I saw that over 30,000 of these records are actually from the Gulf of Mexico. And most of the newer records are from video footage from dives like the one that we're watching right now. So that's pretty cool.
That's really cool.
Awesome.
And, just to put things in context of today's dive, so I know we're going down to 2,400 meters, and we find corals and sponges in the Gulf from anywhere from 50 meters to even deeper than this. So when we're thinking about protecting corals and sponges for different species of fish, Right now, this area wouldn't be a priority for protections from current fishing impacts because there's no fisheries that go down to these depths. But who knows in the future, especially when people are fishing at deeper and deeper depths.
Mm-hmm. These communities are tied together as well, right?
Yeah, and that's true too. We're finding out so much more about how these more shallower communities are related to these deep water communities, and it's just a really fascinating area of research.
I know you guys had a hand in closing some of the marine protected areas in the Gulf. I think that just passed.
Yeah. Yeah, so thanks for bringing it up. I was going to talk about that a little bit. In 2018, the Fishery Management Council for the Gulf of Mexico voted to protect 13 new areas for deep sea corals in the Gulf, and then NOAA officially designated those areas in 2020. And for everyone listening, so these are habitat areas of particular concern. So these are areas that have special protections on them because they have certain characteristics, like they're super rare, because of like their geology and how those benthic communities aggregate on them, or maybe they're particularly stressed by certain human activity. And then some of them are selected because they just provide really important specific ecological functions for like certain federally managed fish species.
Mm.
Yeah, I know. I think part of some of that closed area was, I know from data when I was on the Okeanos, which is super cool for me.
It is nice, yeah.
And I think one of them was actually named after my boss, John Reed.
That would make a lot of sense. That's such a cool example of like just how this type of data collection just directly feeds into those management decisions for protecting these areas.
Exactly.
Hey, Danny, this is Matt. I like to call it the virtuous cycle of data, where we go out and explore and find these really neat things, and Deep Sea Coral Program and NCOS and these different parts of NOAA and other research agencies take this data and use it, and then they bring it, and then we can actually use the information that they interpolate from it to make better dives in the future as well. Wow, we're seeing that's
another really good point.
We're seeing some really cool sponges and corals right now.
Wow.
Yeah, that
white colony, the small white one in the foreground on the lower right, that's definitely one of the coralliids, so the precious corals. And we wondered if earlier the one that had the apocopheran worms on it was another. So there's a live one.
Yep, and we see a couple more of those Farreids as well. The glass sponges that we already collected one of them.
Oh, nice.
Really, really unusual morphology there. Don't think we see that too often.
That a lot of these suspension feeders, the corals and the sponges, they really like being just below the ledge. And I think that's because the current is accelerated there, and if you're on the top, you get the accumulation of sediment. And so it's kind of fun to see the difference in the distribution here, the micro-distribution, I guess I would say.
I think that invertebrate had a vertebra. That's what that Farreid spine looks like. Huh?
Yeah, it does. So, Danny, you planning on staying most of the dive?
Yeah, wow, I was like mesmerized by how cool that was.
I know, right?
Started. I'm going to stick around for the rest of the day. I've been watching these guys on and off, and just it's so much fun.
Yeah, right. I know, it's awesome. I know we had submitted a lot of data to the Deep Sea Coral Program, what was that, last year or the year before, at Harbor Branch. I went through all the records that a biomedical department had collected over the years. I think we have 36,000 collection records. And I went through all that data and all the photographs, and John and I ended up submitting all that. So some of my data is in that. Some of our data is in that, in your massive database. So that's kind of fun.
I've definitely definitely seen it in there. And that's another thing that's like so cool about the database is that like anyone can go on our website, deepseacoraldata.noaa.gov, and download it. And there's images from these dives associated with every single observation we have in there. So you can even like get the URL and look at the specific point in the dive where we observed a certain specimen. So that's so cool.
Yeah, and I know Harbor Branch has been also working on getting all the video clips of everything they've collected up, too. And I don't think that that's public yet, but they've been working on that behind the scenes for a couple years, where we've got people to go through all of our data and all the video clips and identify each and every single one of them that we've had. Because a lot of the stuff they had was well before the technology that we have today, but they were using the submersible. But they didn't have, like, they were using film and Polaroids at one point. So they digitized it all and then took the video footage, which sometimes is on, like, VHS, and digitized that. And then now I think it's searchable at this point. It should be online soon. So we have something sort of similar, but it's more than just corals and sponges. And also it'll be a duplicated effort because all of our corals and sponges went into your data set. So that's kind of cool. And then the other thing I think is awesome is I actually did my master's degree, my thesis on the biogeography of the Straits of Florida. And it was well before we had any of this digitized. And I actually had to physically go to the Smithsonian and do my searches on their computers. And then I had paper copies of what the Gerda had picked up in the 60s and 70s, or whenever that cruise was, and I had to just go through every line of data and collect every single critter, and then I mapped the biogeography. So you guys have done a huge advancement in my little tiny, what now is a tiny data set. It was before worms, before we had a road register of marine species, so I had no way of checking the data set back to real taxonomy without going through every single line. It was very taxing. Did you hear my joke? Badooch.
Steph, it makes me really happy to hear that. Sorry, that Harbor Branch is preserving that legacy.
Oh yeah.
Like to lose that data, like that's a mountain of incredible observations.
Absolutely, 35,000 records they have picked up, and they're all in jars at Harbor Branch. You can actually rent them. Borrow them like a library, take out the samples. I think you can do that at the Smithsonian, too. They'll let you borrow. That's what you're in charge of.
Yeah.
Yeah.
John?
Stephanie, this is John.
Hey, John.
Hi. So, yeah, that database, it is online.
Oh, good.
Public now. And we're going to change the name of it, but right now the website is called shinyapps.fau.edu.
Awesome.
SHINYAPP.
Oh, I love it.
.fau.edu. And in that database, it's kind of like what Okeanos is doing, but it's every sample we collected from 1984 to the present is in the database. There's a world map, so you can zoom in and say, I want to see what was collected in the Galapagos Islands or the Eastern Atlantic or the Caribbean or whatever. And then there's a thumbnail for each sample, an underwater photograph. We had one, a thumbnail, and you click on it, and it's a full-screen view. And if it was collected with the JSL, we made a 20 to 30-second video clip of that sample. It's also linked to that specimen number. So you can click on the video and watch the live video. And it's just like, what you're doing with the Okeanos. This is the first time many of these species have ever been seen in real life. Mm-hmm. So many of them, especially the deep water organisms, are known from museum samples and dredge samples from 1800s, as Stephanie pointed out earlier. And for the first time, we're able to see what this animal looks like in life. Its real color, the habitat it's in, and then the deck photo and so forth. And so we're putting this out so that So the data and the samples will be more available and useful for the scientific community, and we're hoping to utilize that much more. The other thing about the marine protected areas in the Gulf of Mexico, I just want to mention that several of the new HAPCs along the West Florida shelf is what we discovered in part during the oil spill.
Mm-hmm.
And that was our last Johnson Sea Link submersible cruise that Stephanie and I were on. And one of the discoveries that Stephanie was in the sub was a deepwater Lophelia reef due west of
Tampa or Naples?
Tampa.
Tampa.
It's kind of just slightly north of where you guys are now, 500 meters of water.
Mm-hmm.
We call it Stephanie's Bump. It is a beautiful, very thriving Lophelia reef that is now part of that HAPC, along with some other parts. So, yeah, great work.
Thanks so much, John. I'm glad to see John's legacy solidified in data, publicly available. You can see all the work he's done for 40-plus years at Harbor Branch. When I first went to sea in, I was like 2005 with Chuck Messing. He was my master's professor. I met John on the boat, and Chuck said to me, "This is John Reed. He's like the king of deep sea corals," and something like that. And I was like, okay. And I met him, and I was like, I'm going to work for this guy. I am going to work for this guy if it kills me. So I got hired in 2010 by John after I graduated grad school, which took a ridiculously long time for me to graduate because I did not have worms, and I was doing a humongous metadata analysis that took forever.
Yeah.
Yeah. So I've been working for John ever since. So he's threatening to retire. I don't know if he ever will. But, yeah. But thanks for chiming in, John. That was awesome. Also, I just tweeted the location. And, Danny, if you give me the Deep Sea Coral link, I'll tweet it for you, too. If you follow me on Twitter, it's at Deep Sea Nerdy. I just tweeted our Harbor Branch data portal out. So you should be able to just go to my feed and find it. Or if not, if you don't like Twitter, it's shinyapps.fau.edu. And, Danny, if you can repeat the name of the Deep Sea Coral website. Go ahead.
So it's just deepseacoraldata.noaa.gov.
Awesome. Yep, it comes right up on my phone. I should have known that. Thank you. So I'll have that up in a second on Twitter.
Another piece of this data puzzle is the annotations. Like someone actually going through and putting this information in, and Scott's lab does a lot of that for the NOAA Ocean Exploration Program. So we're grateful for Scott and his graduate students putting in many, many hours to review this information and put in their best possible classifications for these animals that we see, and then that filters in to the Deep Sea Coral database as well.
That's nice. That large Antipatherian.
Yeah, that was a beautiful specimen, really tall. So I imagine it's pretty old. I'm not certain if that's a Bathopathes or a young Telopathes, but I'm gonna lean towards the genus Bathopathes.
Mm-hmm.
So that large anemone we were looking at was reminiscent of an anemone that's called Relicanthus, and it's a really interesting case because it's not actually a true anemone. It looks a lot like an anemone, but it's its own sort of evolutionary branch within the hexacorals. That one looked a little like its tentacles were shorter than I would have expected for Relicanthus, but I don't think we know a ton about them, so we don't know exactly how they grow.
There's this nice white fan out there. Bamboo coral? Maybe? Really fine. That's beautiful. Any ideas, Scott?
That's another one of the coralliids.
Coralliid. Thank you.
Note the way, when you're looking at these, the way the polyps come off of the branch, they almost look like they are opposing each other. So little barbells or little dumbbells. And then very nice sort of fans, and often the polyps are sort of oriented on one side of that fan.
Awesome.
They're good markers to identify them.
Got a lot of stuff growing off the bottom part of it where it's maybe dead.
Yeah, that's interesting.
They have very solid skeletons, so the skeletons persist for a very long period of time, and so you often do find lots of other associates that have colonized the skeleton after the tissue of the coral is gone, even though parts of the coral, the original coral, may still be alive. And you may have heard me earlier refer to it as precious corals. This family is the precious corals because the shallow water ones in the Mediterranean have been harvested for more than 1,000 years, couple of thousand years, to make jewelry. So the pink coral jewelry is in this same family, Coralliidae. And that skeleton is so hard that it can be basically polished, which is the appeal.
We're going to stop to take a little look at this tall, thin sponge that's baffling me.
This is one of the ones we collected while I was at lunch?
No, we collected a farreid. I don't think
this is something different?
Well, maybe I'm not seeing it. Oh, yeah. I guess it is.
Is this
yes,
very,
it is. It is a farreid.
It's very
it's just
consistent.
So narrow. Those flutes don't spread out much at all.
It's so weird.
Oh, and then again, there's that black coral that we saw before with the concentration of polyps into its central axis area.
Which I have forgotten the name of already.
So have I. Heteropathies,
That's
I think.
it.
Yes.
That's an easy name,
Thank you.
too. You think I'd remember that one.
Either good memory or good notes.
Yeah, right.
It's heteropathies, or there's another genus called hexapathies, and we'd have to wait to see one where we can see it, the plane view, to see how the branches are actually arising from that central stalk, to know the difference between those two.
I see a lot of stalked crinoids up here, too. We don't get to see this very often in the depths I usually work in.
Oh, yeah, there's that black coral. You want to zoom in on that or just keep moving?
Yeah, sure. I thought, did we not zoom in on already? I thought they saw it already. Oh, yeah, go ahead. Snap zoom then.
Quick look, yeah.
One thing to note about these, this particular Bathypathes, so it's got that central axis, and then the side branches, they're referred to as pinnules in the black coral taxonomy literature. And note that they're all about the same length. And so when you zoom back out, this kind of looks like a big rectangle, this whole colony. And that would be one characteristic that could be used to ID it. And the other would be if these pinnules, the side branches, are coming off directly opposite one another, or if they alternate somewhat.
Right. So Bathypathes alternata would be the alternating one. What's the non-alternating one?
This is non-alternating, right?
Yeah, well, I think they're both in the genus Bathypathes, but there's a species Bathypathes alternata, and then there's several species of
ones?
Bathypathes, so
Okay.
I'm not qualified to tell you which one it is there.
I don't think this is alternata. They look totally next to each other.
Yeah, just the slightest offset. Or maybe none at all. Oh, no, they're just slightly offset. The ones on the right are a little bit higher than the ones on the left.
And this is a really good view to see. The last point I was going to make is whether there are polyps on the main axis or not, and there definitely are.
Yeah.
In those pinnules. Nice. Great view.
Excellent. Thanks very much.
So bizarre. It looks like, there's only 2 tentacles per polyp, but again, there's not. There's 6. They're just really stretched out along the pinnule.
You can really see that crust where it's sort of broken away.
Yeah. A little bit of sedimentation on the top parts. How straight these rocks are locally. Like the cleavage on them is really flat.
Somebody's going by above
What's
us.
up top? What is that? Ooh. Ooh, a squid. Ooh,
Oh,
ooh,
yes, a squid.
Ooh, follow it. Oh, gosh.
Oh,
I know these. I've seen these before.
I know. I've seen it as well. I'm thinking,
With the bent arms. Look how cool.
I know, it's just so...
It's a big fin squid,
it's
maybe?
ghostly.
Magnapina?
Magnapina?
Oh, man, look at him. He's very alienish. Ooh,
Look,
how
you can see right through its...
No. Mantle.
Right through it.
And through its fins, look how beautiful.
Incredible video and ROV work here.
I know. Look how long his legs are.
Wow.
I would imagine those two extra long ones are the tentacles. This is just me guessing at this point. If this isn't
Hi,
Mike
Okeanos.
Vecchione, thank you. Hi, Mike. Look at him go!
Hi. Yes, that is a Magnapina. I actually thought I saw one yesterday, way back in the distance, but you never got a
close
good
up
look at it. But,
Yeah.
I've been waiting for these to show up on this dive because you're pretty deep now. Magnapinidae is a family of squids that Dick Young and I described back in the late 1900s.
Nice.
And used to be easy because we thought there was only one species, but it turns out we've now found at least 3 or 4 species of them.
Awesome.
So as far as your logging is concerned, Magnapina SP.
Yeah, awesome. Thank you
Excellent.
so much.
Great job following that.
And there was a recent paper came out from Australia where they saw, I think they had 5 observations, and they figured out how many observations there had been published up until last year, and it wasn't very many. But Okeanos runs into them every once in a while, particularly around the Gulf of Mexico.
So cool. Well, we got one more data point.
Was so great. Exactly.
Yeah. It was a really nice video.
So cool.
And a very popular subject. People really like the big fin squids.
Yeah, they're really interesting. They're so odd
Great
looking.
how we got views of it from almost all the way around
Yeah.
it.
Yeah, they are very odd. Do you have any other questions about them?
Are they the same kind? Like you were telling us the other day, how they hold their legs, like in like a wrap. Is that what I was seeing? Yeah. Is that what I was seeing with him too?
No, they're related to that family. That was referring to the family Mastigoteuthidea.
Okay.
Where they have 2 elongate arms and they hold their tentacles in a groove in the ventral arms.
Mm-hmm.
But the Magnapina, they have all of their arms and tentacles have this extension on them, long spaghetti-like extension. And so they have to hold all 10 appendages out. And it's really difficult to tell the arms from the tentacles, which is very unusual for a squid.
Yeah. I thought
they have to hold them out away from the body so they don't get tangled up.
Oh, geez. Getting a rat's nest of tentacles in his body. I thought it looked like there were 2 really long ones versus the other. Like they looked different.
All 10 of them can be extended very long, very long way. They've been the longest one we found was reported to be about 21 feet.
Nuh-uh. That's crazy.
Yeah, with just a body that's just about a foot long, but the,
Oh my
the
gosh.
arms and tentacles extended out almost 20 feet from that.
That's amazing.
Did we ever have the lasers on this guy? I feel like
I
not.
don't know. I don't know. We
We're
can look
just
back
too
at the footage. I
mesmerized.
know.
It's so funny because we
We'll go
just
back
saw
and
those.
find it again and put lasers on it.
Yeah, right. Speed back. Speed back over there.
Yeah.
Turbo button. Thanks for chiming in, Mike. That was awesome.
Got any other questions?
I'm sure I'll find some.
Before you go.
Wait a
Mike,
minute. Sounds like somebody else.
Scott's talking to you.
Somebody's got a question.
Yeah, Mike. So I'm not sure how big that squid was, but how big did these get?
The biggest one that we have reported was about 21 feet long. It was actually the first one that I ever saw. And when I was contacted about it, the person contacted me, said, "We've got video of a 21-foot squid. Do you want to see it?" And that was before we knew about this family. So I thought, sure, that it was a giant squid. So I said, sure. But then when I got the video, and it was completely different from any other squid known, it was a lot more exciting than a giant squid.
Architeuthis is
We can call them
nothing.
instead of giant squid, we can call them pretty big squid.
Yeah, except that the body part's not very big at all. It's just that they've got these long, gangly arms and tentacles.
Thank you.
Sure. Okay, I'm going to hang up.
Okay, thanks. Thanks for being there, Mike.
Bye. Sure.
So we have made it to about 2,377 meters up this ridge, which we dubbed Shark Fin. And this NOAA Ocean Exploration EX2107 Dive 10. It's all good. Now we still have that holothuroid showing up.
I'm looking at the replay. Our video clipper sits next to us, and there was lasers on it.
Oh, good.
So, yeah, someone can figure it out.
More wall up ahead, more rock.
So it looks like we're on the sandy top up here, the top of our slope. Probably about a 20-meter ridge. That's what I'm looking at. We've got more sedimentation on the top with bioturbation. Yeah, you can see a really good view of it on Sirius. Oh, I see. It drops back down again. And then it comes back up in the background. So it's probably closer to like 50 or 100 meters wide. I think I see a sponge, a little white sponge on those rocks over there.
Yes.
Maybe Hylanema? What do you think,
To me,
Alan?
from here, I would guess that's, a little stalked glass sponge, hexactinellid, Hyalonema.
You kind of see him blowing around in the current a little
Yeah,
bit.
it is moving a little bit, so it is stalked. But as I mentioned multiple times, stalked hexactinellids are present in many families. But
about 10 centimeters on the bulby part.
There we go. It has such a fine stalk, really tiny. Yeah, I still think that mostly looks like Hyalonema. Very nice. Great view of it.
Just when you think you're done, I give you a little bit more.
Ooh. Really pretty.
I'm not sure. Let me check with Nav. Nav, is this science?
Thank you for that zoom.
Yeah, the crinoid at noon. Look at that. It's a beautiful stalked crinoid.
Gorgeous, really gorgeous.
He's probably the biggest one we've seen, I think. So this is related to the sea stars or starfish or ophiuroids, we say. It's an echinoderm.
Yeah, and the holothuroids.
They're also in the fossil record. Crinoids were pretty common, yeah. Even in the shallow waters. Now they're just restricted to the deep at this point, the stalked ones at least.
We're getting a nice view down into its mouth region. You can see how those arms with pinnules collect food, and then they transport that down to the mouth.
Do you want to do a water sample here, or you want to wait until the end?
We have 2 to go, and I would say when we're really in a dense area again, and then at the end.
Okay.
So Mike Vecchione in the chat is reporting that they just had a paper accepted today on Magna Pinna, and as the first observation of a squid at hadal depths.
Oh, nice. Back in the sandy area, the top of this ridge. Lots of bioturbation. I would imagine the sand here is pretty thick. At least it's thick enough to have infauna. There's some more hard bottom ahead of us. And already I see a long stalk at noon. A small shrimp at 3, well 4. And another one of those purple ophiuroids. I'm sorry, purple holothurians. There's sea cucumbers. Maybe some more Hyalonema stalk sponges in the sediment. See 3 or 4 of them. Really short. That one's different.
Ooh, yeah, that looks like Farinema. Yeah, I think that's the word I'm thinking of, Farinema.
Farinema?
Let's see.
It's very hairy.
Yeah, they're very
long
hairy.
spicules.
That's how they hold on to the bottom. So we haven't seen this on this expedition yet.
So they're typical sediment dwellers, or
Yeah. They're typical sediment dwellers, and they have this furry bottom. Thank you for that.
It's even Movember in the deep sea.
That was P-H-E-R, Pharonema.
Okay.
Now this piece looks like it's out of place, right?
Mm-hmm. Like it's about to slide off. It's probably a single-stalked bamboo coral. 9 o'clock.
Mm-hmm. Seems like it's quite a bit wider right where we're at now.
Mm-hmm.
Some sort of saddle. I keep imagine trudging across this, like if it was exposed, climbing up the.
Like a mountain climber?
Yeah. Climbing the ridge.
Yeah, it looks like some of the rock formations out in California.
What's that off in the distance there, sort of straight ahead?
The white
Maybe that's
thing?
another Pharonema.
It's quite sandy up here. I was expecting it to be a lot more rocky.
It's another sponge for sure. Guess it is.
Yeah, it looks similar with that, like really hairy.
Thank you.
Maybe a bunch of amphipods or something in it.
$23.73. Still got about 70 meters to get to the.
To get to the lowest point?
Yeah. I think, though, if I recall the way it's shaped, though, it goes up to a point and then goes down and then
yeah, then we might be in that
goes up
settled area.
again.
Mm-hmm.
Yeah.
I'm not sure how far we've traveled as far as like horizontal distance.
See, our chat room got a little quiet.
As did we. Taking a break.
Yeah. Well, and this saddle is just more sedimented than we had thought. And unlike our one dive, you know, just a bit off the bottom, we're not seeing a ton of the water column creatures here. Just the one awesome Magnapinna squid.
Right. That was pretty cool.
Yeah.
Got something there at center screen. Maybe it's just rock or something. I don't know if it's fauna or not. There is a Bathypathes. Oh, it's rock. But there's Bathypathes down at 7-ish. Yeah, quick zoom on that. That's good. Yep, that's a black coral. Okay, we're good whenever you guys are.
So that's a little different than the Bathypathes you were seeing before, right?
I'm not sure. Yeah, the other one was really square. This one's more rounded, I guess.
Well, this one, I think you said it, Stephanie, the other was more square, rectangular. This one has the tapering branches as you move to the top. So not all of those side branches, pinnules, are the same length.
Yeah, so that's probably a different species than we saw earlier.
We have some requests to try to measure the size of the one that we saw before that's more sort of rectangular looking.
Okay.
Size and
I would imagine we have lasers on it when we saw the one before. So we might be able to go back and footage and look at that. We definitely can't turn around, but that would cause a big drama.
No drama needed.
Yeah.
The request was if we saw it again.
Yeah.
That last one, I think, was a much more classic Bathypathes with the tapering branches and the curvature to the whole colony, like a feather.
Yeah, really feathery. Is there a fish up there? There's a little shrimp. Yeah, okay, so I'm actually looking at this, let's see, 10, maybe a meter long. So I'm sorry, my brain's not working anymore today. Caitlin, who's doing the video clipping next to me, is showing me the footage of that Bathypathes we saw earlier that was very rectangular. And it looks like it's almost a meter long, maybe 40 to 50 centimeters. I'm sorry, half a meter long. Half a meter, yeah.
That'd be half.
Half a meter, sorry.
But it's also difficult to tell from that angle. Oh, I see. More critters are coming.
That's odd. Can you pan down a little and look at that sponge in the sediment right there? It looks like an old hat.
Oh, from far away. Yeah.
Yeah, same one, right? With a really hairy...
Yeah, absolutely.
Yeah, that's what I was thinking, too. The sorting hat. Looks like the sorting hat from Harry Potter.
Better be sure. Nice. So that definitely has that Pharonema look to it.
Ravenclaw.
So, my daughter is about to be in the show Puffs.
What's that?
In a community theater. Oh, it's sort of a takeoff on Harry Potter.
Oh, fun.
All focusing on the Hufflepuffs.
Oh, no way. Yeah. That's awesome.
Yeah.
Do you know what you've been sorted into? Have you taken the test?
Me?
Yeah. You've never taken the sorting hat test? They have a couple
no,
of them
I've never
online.
taken the online test. Yeah. But I'm pretty sure it would be
I think it's on their website.
Slytherin.
Oh, yeah, you think?
Mm-hmm.
There's not to witch a wizard who's gone bad who wasn't in Slytherin. I think I'm a Ravenclaw, if I remember right.
I can see that. probably I'd be lined up for Hufflepuffs. Third or nothing. I'm glad we got that second look at that Pharonema. You could see all those, they call them basalia, the basal spicules.
The hairy part.
The hairy part.
The hairy
They were
nest
so
itself.
long.
Yeah, they're neat. Ooh, I see a fan. I see a fan at 3 o'clock.
Yep.
Off in the distance. Oop, something in the water. Hatchet fish? What is
Not
that?
a hatchet.
What is that? Shrimp? He's going nuts. Ooh, come back down.
Oh, well. So we're actually making quite good progress. It's about 1:20.
Do you know what our off-bottom time is? Make sure we're going further enough. Let me check with Nav. Do you know?
I'll check it
Okay.
in the plan.
They're coming up on the sea fan. My guess from here is going to be bamboo coral. Yeah, I see the nodes. It's very soft.
Off bottom time says 4 o'clock.
Okay. I thought we had a really long descent. I'm surprised it's so late.
That's making me question whether that's true. Ooh, look at that bamboo.
This is a nice one that we have not yet seen today, or at least we haven't identified it if we've seen today, but that is awesome.
Yeah.
This suggests from here that it would be provisionally in the genus Isidella, which case it would have branching at the nodes. And I'm basing that on the thickness of the branches, the height of the polyps, and then it's almost like V-shaped branching, the angle of the branch coming out of the main stem. Very nice. So obviously a really big one if we can see it that well from this distance.
Yeah, it's large.
With another one over the edge.
And that one looks different.
It does. That looks like that Corallidae. Hemicorallium, I think. It's mentioned in the chat somewhere.
There we go. Oh, it looks like the branch breaks right above the node.
Very interesting. Yeah,
That's neat.
I agree with you.
The polyps look a lot like those zoanthids we see, but they're much more organized. With a really wide, flat mouth. Very fine, thin tentacles. It's way different than the first one we collected, which is like a similar size body for the polyp, but way longer tentacles. And it was much more disorganized than this. This is really organized and planar. From the angle I'm looking at it, I don't see anything really making a 3D, like any branches going the 3D direction, more than just 2D direction.
Yeah, I think they would refer to this as like a multiplanar,
Okay.
multiflabellate colony. You have multiple planes that may come out from the base. Really interesting. I think it's worth noting again the ecology of the distribution of these coral colonies. You notice they're all over here on the edge of this ledge that drops off onto the sides of the ridge crust that we're transiting up, and they're not in the center of the ridge crust. And of course, we can see that there's an awful lot of sediment here on the ridge crest in this particular area. And so, A, the hard bottom is being covered where the larvae would have to settle and get a toehold, but B, that sediment accumulates, and so if the coral isn't growing fast enough, it doesn't basically grow above the sediment or stay above the sediment. And so these edges where the sediments fall away are the better place to settle and grow, and the currents get enhanced sweeping over the edge of it, and so there's more food delivered per unit time. Really nice demonstration of that here.
It looks like a couple species of coral coming up.
It's great, Scott. Thanks for that. I think that one in the center that's very semicircle, that's a Corallidae again, you think?
That's probably. It looks like the biggest one we've seen so far.
It's just so perfectly formed.
That guess.
Yeah. And then bamboo corals around it. Yeah.
So much healthier than that first one we came upon with all the apelocophrons infesting it, for lack of a better word. Sorry, Kevin Cosett.
Yeah, this is a nice view of this to, I think, verify that that's what we saw earlier. Maybe one of you just mentioned that you could really see the zigzagging in the branching pattern here.
Sorry, we're doing drills today, so if you heard the bell, everything is okay. It's just a drill. Nice. And I also see some crinoids maybe at the top. A stalked crinoid up there?
I think we should keep going upwards. Thank you. Just keep an eye on this edge as we go.
Purple holothurian. Ooh, little fishy shrimp. Looks like similar to the one we saw earlier. Stop moving, bug. Sit still. There you go. Ooh.
I'm just wondering if that's an amphipod.
Yeah, it might be. It's very, I think it's an amphipod.
Yeah.
Very robust in the
chest region.
Yeah, chest area. He's been lifting his weights.
It's upper abdomen.
And some more of these holothurians. Holothurians are definitely the most common species out on the sediment areas, at least that I can see. The ones that are not buried in the sediment.
Yeah, I think if we can hug the right edge where we're seeing so many corals, we can keep. Excellent. Perfect. Thank you very much.
Yeah, it's really bioturbated up here. Lots of tiny little holes and mounds. Maybe some feeding tracks.
We've never seen that orange holothurian again. The one that looked like a tongue? Was this?
Oh, we saw two of those.
Pink.
Yeah,
Pink.
two of the pink ones. That here's a light pink one, but I think it's a different. The other one, the tongue-looking one, wasn't really clear.
I'm not sure we've seen this one. Maybe we should take
we
a little
zoom on
look at that zoom. Chris Ma was asking if we had gotten a good zoom on. Oh no, yeah, that's different, right?
Yeah, I think so.
So we haven't taken a close look at this one yet.
That's neat. Really lumpy. He looks kind of tongue-like too. It might be the same genus.
Mm-hmm.
Or similar.
There.
Basically a crawling intestines. They take sand up in one end and dump sand out the other. Collect all the
yeah, this is a synallactid as well.
Organic matter out of it.
Great. I think that's good. Thank you.
Mm-hmm. He's cool. So according to our abandoned ship drill, we're 106 miles from land.
106?
Yeah, that's what she said, I think. Even though I have 146 on the wall. Maybe I misheard her. So it looks like another Paramuricid. And the Corellium.
I think we collected that Paramuricid. Paramuricia.
Yeah, let's have a quick look, see if this one also does not have an ophiuroid. I'm not seeing any brittle stars on any of these colonies today, which is kind of interesting.
Yeah, me neither. Is it too late? Can you guys turn to the right and look at that yellow gorgonian that we passed? If you can't, don't worry about it.
Yeah, you don't have to go back. That's okay.
Yeah.
We're almost surely.
Yeah, it's right there.
But I mean, not on any of the corals up here. Do I recall seeing any of the snake stars? I remember a dive like that before where we didn't see any of the associated brittle stars.
Yeah, the yellow one.
I don't recall if that was down on Toe Ridge or it was elsewhere. Yeah, nothing on there again.
Thank you. The yellow one next to it, we were
Just
looking
to
for
the
the
left.
ophiuroid, which it doesn't have. So, yep,
Which it doesn't have.
He is ophiuroidless. Very good, thank you.
See, got some good barnacles in there.
Also saw some zoanthids on this live coral on the left, right? Yeah, we're good. Maybe our second to last water sample. Let me check. Hold on. Navis' science. Do you know what our off-bottom time is? $20. Sorry, what? Oh. No wonder I'm confused. Thank you. So about an hour and a half.
About an hour and a half? Okay.
Yeah, so it's closer to 3 o'clock than 4, which makes more sense. Okay.
Yeah.
We got some neat corals up here.
Great. I get it now.
Ooh, maybe a tripod fish? We haven't had very many fish up here.
No. I think that's that Viridensis.
Cool.
Oh, maybe not.
He's definitely up on
the
his
black-headed.
stalks.
Yeah, I think that is that one. Well, is it that one?
No, look at the...
No, yeah, you're right. It's different than that. So
Bathyopterus species. I like their perpetual smiles. They always look so happy chilling there.
I know. I really like these fish.
It's so neat. Cool blue head. It's really faint. A tiny little eye. And then they just stand up on their stalks. Their modified fins.
Waiting for prey, sensing the prey coming along. Yeah, I'm not sure which one this is.
Ooh, is that a little parasite?
One of the ones we saw yesterday had parasite, an amphipod parasite.
I don't know what that is. I don't see legs.
No, it's nice we have a good close-up of it, though. We can
Maybe it's
share that with people.
Looks kind of segmented.
If it's an egg sac.
Egg sac? Could be an egg sac.
Somebody laid its eggs. Well, that's another name for most of the parasites, right?
Yeah.
Yeah, well, that is true. I was thinking more like a little shark egg sac or something.
Huh.
I'd be surprised that the fish would tolerate that. Unless it got caught on its way by or something.
Oh, it's on it. Oh, I'm sorry.
Yeah.
I looked away and I thought we were looking at the fish. I thought we were at the base of a sponge.
Oh.
Yeah, sorry.
Like,
that's some kind of little crustacean parasite.
Yeah.
Assuredly.
a worm
This
or
is really,
something.
you know, another indication of exactly which way the current is going.
Mm-hmm.
Some comments.
Dion suggests it's a leech.
Yeah, and Megan McCullough as well. So. Have to run that by a parasitologist. I happen to know one.
Oh, nice. Look at this, like spiral looking circular bioturbation here in the corner.
Mm-hmm.
It's kind of neat. There's lots of shrimp. You see 4 or 5 and 4 or so in this view. Little shrimp that are on the bottom.
It's great how you can see the shadow of D2
Mm-hmm. Oh yeah, from
the lights on Sirius.
Above, yeah. Nice ledge here.
Here we are getting our another peek over the edge. Now it's not as sharp at this lip.
Yeah, it's rounded sediment top, and then a drop off.
And there they are, all the fans.
Nope. Maybe a water sample here where all the fans are?
Yeah, I think maybe that's a good idea. If we could make another water sample just up ahead, sort of, where we know the water's flowing through these fans. Yeah, like just a bit ahead. 5 meters or.
This Faria was the last thing we collected, right? Number 7?
Yes. So we're 8W. Niskin 4. eDNA. So it'd be interesting to take this water. So one of the interesting things that you can do with an eDNA sample is you can use what we call a universal primer to try to amplify a set gene that will amplify as many different organisms as possible. So if you could, this is perfect. Yeah. Success? Great. Thanks. So I was saying that, you could use a universal primer, something that amplifies a gene from, say, all animals or most animals, and then you get a snapshot of the community that whose DNA is represented in the water. But you can also take these samples and then say, well, how about an octocoral specific primer, and you amplify out of your eDNA sample just using primers that should only amplify octocorals, and then you can have a more focused look at a different section of the community. So the eDNA samples have a lot of utility. You can look even for a single species. So that one of the things that people do with environmental DNA samples is they try to detect a species of interest, whether it's commercial or protected species or an invasive species, for whatever the interest is. And so you can use these environmental samples in multiple different ways. And so when we take these samples and deposit them back in the Smithsonian National Museum of Natural History, we'll do some sort of basic community profiles, looking at as many different organisms living in the surrounding water as possible. But then also, they'll be part of the sample will be archived, and other researchers could use them in the future to address specific questions. Yeah, I'm glad we got that sample right there where we could see all those different fans.
Maybe some human debris there?
It looks a little too rectangular for...
That is a human debris. Metal, maybe? Got a little anemone on it.
Anemone, yeah.
Could be metal. Looks almost like a piece of flooring, maybe? Wood flooring?
It's like a panel.
Mm-hmm.
Well, it's nice that this is the only human debris that we've seen on this.
Yeah.
And that anemone said, "Thank you very much. I'll make this my home."
That's right. Nice solid place to put his butt.
Huh. We got a comment on the parasite on that tripod fish.
Okay.
Alistair Dove, this is via Twitter, says that it was definitely a leech and guesses that it's a piscicolid leech. Thank you for passing that along, Megan. That's really great. One of the fun things about this whole exercise is, we're narrating what we see live, but also there's a whole community of people out there who are taking snaps of what's shown. And, so Steph and I know what we know, but we don't know a lot, right?
We don't know
so
everything for
a whole
sure.
bunch of other people can use this video footage and help basically serve the aims of the exploration to increase knowledge about what's down here.
And it makes my life easier. So I'm not sitting here with books upon books, digging through papers, trying to figure out what stuff is.
Yeah.
Which is what I would normally be doing if we were out by ourselves without the telepresence.
Yeah. So I think that's just a great example of how this all works.
Yep.
Oh, is that Bathypathes is a little more rectangular?
I think it's the rounded one.
That's, yeah.
It's more like Christmas tree shaped, right? Like
Yep.
pointed at the top.
I can see that. Yeah.
That one, the branches really do appear to be alternating, so this looks more classically like the Bathypathes alternata. Actually, as I'm saying that, I'm remembering, I think that Tina Molotova in Russia described a new genus, Alternapathes.
I thought they were synonymized. That Alternapathes and Bathypathes were
one or the other.
synonymies.
Yep.
I don't know.
Usually worms keep, World Register of Marine Species keeps those organized.
Oh, this is cool. It's narrow. This ridge is really narrowing.
Still heavily sedimented on top. It's just steep drop off on either side of it. That's basically what we expected, though, based on the multi-beam. That's why we called it the shark fin. Really thin dorsal fin-looking rock formation. Got a little shrimp right at laser right now.
Just to follow up on that Alternapathes comment, looking at the Benthic Animal Guide, the images of the genus Alternapathes look very much like a drooping palm.
Okay.
Combined with a feather, so I don't think that's what we're seeing. So these can just stay as Bathypathes.
Okay. Sounds good. Pretty barren other than the random shrimp out here on the sand. Sometimes we do see the holothurians. And this heavily bioturbated sediment, which is indicative of having in fauna, likely worms or clams or other gastropods or mollusks in the water. I'm sorry, in the sediment. We can do a mid-dive rundown, I guess. So
sure, why not?
If you're just tuning in, or you've been tuning in for a while and we haven't given you an update, which I think is correct, then we'll give you a quick rundown of where we are and what we're doing today. We are 76 miles from the Dry Tortugas and about 146 miles from Key West, off the Keys. We had to leave the Blake Plateau, which is really the target of our expedition this month, because the weather was too bad and the sea state was going to get insane. So rather than all be vomiting everywhere, we escaped to the south and southwest, and we are hiding behind the Keys right now. So we got on a couple dives yesterday. We did something in Tortugas Valley, which was it was very sandy with like this like clay-like rocks when we did see rocks. And here today we're on a much deeper slope with this very distinct thin ridge. We're actually on the top of this ridge, running along the top of the ridge, and we started our dive roughly at 2450 meters, and right now we're at 2,358 meters. So we've come up quite a bit. The base of the slope where we landed was still pretty high on that mountain ridge. I think all the way down in the base was closer to 3,000 meters. Mm-hmm. So we came in on like middle of the ridge, and we landed on a pretty sandy spot, and then we started making our way up the ridge, and we got more of these black rocks. And it's a really thin ridge, maybe 50 meters wide. And then the top sides of this ridge are really sandy, with thick sand, but on the edges and the walls, it's exposed black rock with lots of coral. It's pretty good coral habitat here. Most of the fauna we've seen on the rocks is a type of coral of some kind. We also have some Hylanema sponges, or the ones in the stalks, little stalk sponges.
Mm-hmm. And an allied form, Pharonema, that has the...
sorting
up
hat.
Yeah, exactly.
It looks like a little white hexactinella and like a nest of hair.
Yeah.
It sits on the sediment. It's pretty cool. Lots of Bathypathes, black corals. We also have seen many bamboo corals.
Precious corals.
Precious corals.
Quite a bit of hemicorallium.
Yep, and we haven't seen those this cruise yet, so they're new to this dive. Only one piece of human debris, which is awesome. Mm-hmm. I'd prefer be 0, but, yeah. One is one. What else we got?
We saw that on the sand, an unusual relative of tripod fish called Ipnops.
Mm-hmm. Right, when we first got him.
Yeah.
Then we also saw a big fin squid at Magnapina.
Can't forget that.
That's right. So we're just making our way across the top of this. The dive will be over around 3 or 5 after 3 or something. We have a long descent and a long ascent because we're at such a deep depth that it's a pretty long in and out. What else you got?
Just that we've made some collections.
Anthony,
yeah.
And I just want to point out, I think the Paramaricia here have brittle stars on them.
Oh. All of a sudden these Paramaricia, that none of which had a brittle star before, now we see some.
Can we see the yellow gorgonian at 7? Yep.
Now we have collected one of what looks to be a similar species.
Here we go. He's got one.
Yeah.
And these look different to me. The yellow looks a little bit different color. I'm not sure if the branching pattern's the same, so I'll bet they're different species.
Mm-hmm.
Yeah. Is this something then that we would like to add, since we've collected another Paramaricia that didn't have the brittle star, and this is one of the Aspire targets, two taxa targets as well. The Paramaricia with the Astrashima.
We have room. I'd be happy to get it if they feel safe. Kind of on the edge.
There's a closer one to us, right? Maybe if we can get a closer look at that one.
Okay, thanks.
Right in the center now.
Yeah, they're at the ROV. Settle that real quick. They're going to try.
Snake stars, they call these. Associated.
Astrashima, is that right?
Mm-hmm.
Yeah, that
that's
one's fine.
perfect. We just want the star as well as some of the colony. So
Probably the way they loop their arms, you're going to have to clip the main axis of the colony below the star.
Mm-hmm.
Yeah, you're going to need your Gorgonian clippers for this. And we're really wanting to get the brittle star, so if you can cut the Gorgonian below the brittle star. Yeah. Yeah. I'm going to call the Paramyrceid sample number 9 and then the brittle star its associate, Alan. Does that make sense?
Yeah, I think we should do that. Sure, this will be helpful for a lot of people who are working on this group and working on genetic connectivity studies. Yeah, these polyps look quite a bit bigger than the other ones as well that we collected.
They also kind of seem separated, like they almost
Yeah,
have like
the
either
more open space between them.
Yeah, and they also like even the polyp is almost looks like it's sitting on tissue or wrapped in some kind of tissue at the base, or it's just not as clear, right? See through.
Yeah.
Yeah, these sorts of what we call holaxonians, where all the sea fans, the Gorgonian sea fans are, their polyp structure is such that it's divided into two, and the lower part is kind of like a cup that the upper part that has the tentacles in the mouth can completely retract into. And so one of the earliest colonies that we saw today of this type looked like it just had a whole series of mounds, and that was that lower part of the polyp with the tentacle part retracted into it. And that's different than, for example, the bamboo corals, the primnoids, and the chrysogorgids. None of those have tissue that's thick enough to allow the polyp to retract into. So the polyps can contract, but they can't retract into tissue like this one.
Like its own little house.
This is great. It's perfect. Absolutely perfect.
Oop, popped right off.
Got the whole thing.
Hold fast and all. So it's going to be sample number
9B I have. Not sure what box are we targeting? Great.
So it's going to be 9B.
to be busy tonight.
I know.
Yeah. We can do some photographing and organizing these samples and getting them in great shape to send back to the Smithsonian, where the Smithsonian will preserve them, care for them, manage them, help loan them out to other researchers so that we can learn more from them, more about their genetics, more about the taxonomy. These particular taxa are chosen so that they can help inform us on how these different communities are related one to the next, and what are sources of genetic material that could help another population rebound if something bad were to happen, like what we saw happen in the Gulf a number of years ago. And also naturally communicate from one population to the next in a genetic sense.
Yeah, how genetically connected they are.
Hmm? Great
Thank
job.
you.
Success.
All right, so we got about another hour left. Looks like another large rock mountain in front of us.
Oh, cool.
Giant rock wall. Go for it. Oh my goodness.
Wow. This straight up and down wall. The ridge is pretty thin here, from what I can see. There you go. Looks like some fresh exposed white areas. Some like totally random rock sticking out, like on that slope there. Yeah, sticking out to the left. That does not fit.
No, he's totally out of nowhere. I don't know what to do with him.
Yeah, maybe it's just tumbled down from above. It's an odd angle for it to be sitting, though. Oh!
There's something odd center screen. Can you zoom in on that? It's like, yeah, right in the center right now. Might just be a rock, but it's tricking my eyes. Yeah, what is that?
Oh,
a sea
cucumber who has stuck pteropod shells all over itself.
And even a stick, maybe? What's he got? Oh, no, it's the lassia. It's a piece of seagrass he's decorated himself with. Pretty cool.
Yeah. Olaf Linus. I don't know how to pronounce this name, but Chris Ma has given us the name in the chat. And we saw this yesterday as well. Yeah. You call me Sir Collects a lot. How about that guy? Although that one was not doing nearly as well as the one we saw yesterday, which you couldn't even tell. You could just barely make it out as a cucumber.
We got something tall sticking up over here, hanging on this wall.
Yeah, when I first saw this, it was making me think, earlier we saw that coiled skeleton lying on the substrate, and I thought maybe we'd see some Ertigorgia. We certainly haven't seen those yet. I'm guessing this is a bamboo coral, so maybe that's what that skeleton was.
Really steep here.
So that holothoroid is named for Museum of Victoria sea cucumber expert Mark O'Laughlin. So now I know how to pronounce it.
There
It's
you go.
O'Loughlinus.
There you go. Okay. Pilots, can we zoom in on that stalk at 3? Thank you. Ooh, we got a
flytrap anemone up top there.
Yeah, the back side of them, underside of them. More of these really fine bamboo corals we were seeing towards the left. This looks like a dead stalk of some kind.
Just covered with hydroids. A really rich hydroid community.
Yeah, thanks.
It's a it's an invisible surfer was on that.
The rocks are different colors here. Some of them are much lighter. Almost a brown. And here's that crazy one that's sticking out of nowhere.
Also with it looks like maybe some dead or live stalks. Not big fans, but...
Well, they're not big fans of you either. Okay, I've been doing this
Fair
too long.
enough. Yeah, exactly.
How long have we been on this boat?
Who's making the dad jokes now?
My espresso's starting to wear off. About 45 minutes left. Try and get as far as we can. It looks like a lot of these highs have these really thick sediment on them.
Yeah.
They must be coming down off the plateau that's to the north, where Pulley Ridge is. You know, it's south of Pulley Ridge. There's lots. Outside of the Pulley Ridge area, there's lots of sand up on that shelf up there.
And we noticed when we were coming through the water column that there was quite a lot of sediment and snow. Marine snow in the water here. So it's probably just a steady diet of it.
Coming off from the coral reefs that are in the Keys. Most of the sand on Earth is made from parrotfish, is that right? I think I heard that. To look that up.
I don't know if that's most the sand, but stuff that Wow.
Okay, parrotfish can produce hundreds of pounds a year, but it's the white sand in the coral reef.
Yeah. A lot of sand comes out rivers is from mountains. Terrigenous origins.
They're really rugged. Rocks are really sharp looking. Not a lot of erosion. Some small broken pieces of rock, it looks like rubble or cobble-sized bits. I collected one earlier.
Mm-hmm. Yeah. Scott France is commenting that, there's a lot of rock available still for colonization, and it's certainly not space limited.
No. A fish. I see a fish at 4 o'clock. Right on the wall.
Oh.
You can see him wiggling around down there. This is our maybe third fish of the dive or something.
Yeah.
Not very many fishes dive. And I don't think I've
That'd
seen
be great.
any rat tails. This one looks like a rat tail from this distance. Yeah. We've seen him before this cruise. I'm not sure the species. Some fresh exposed white rocks underneath him.
Mm-hmm.
He's trying to hide from us, probably. Or hunting, one of the two. Ophidiid? Cuskiel. Hey, who's calling in?
Mary Wixton.
Hey, Mary,
Hey.
how are you?
I'm fine. Just got up from class.
Oh, nice.
Well, thanks for calling in.
Well, let's see if you got some good crustaceans today.
Yeah, right. We haven't seen very many. And I haven't seen any Chrysogorgia, right?
Right.
Chrysogorgia this dive.
Mm-hmm.
Right.
We did have some small shrimp that live on the sand.
Mm-hmm.
Maybe
Anything
the
else?
Nematocarcinus, maybe? Or they're pretty big shrimp.
Quite likely.
Mm,
Yeah.
That sounds
well,
likely.
I mean, you can get juveniles as well as adults.
Yeah. Yeah, we'll keep an eye out for you and yell for you when we see them.
The nematocarcinids have extremely long legs that work like stilts, so they do very well on muddy or sandy substrates because they don't sink in.
Okay.
Aha.
We'll get a good zoom on them when we get back on the sediment. There's like this ridge of
All right.
sediment on top that they hang out in. Right now we're just hanging on the wall. Yeah.
Mary,
On the wall.
Yeah.
did you happen to see the shrimp that were in holes in the rocks yesterday?
You had some Acanthocaris, the lobster that has the very long appendages.
Aha. Great.
And the one picture I saw was a really tiny juvenile. The big ones, they were photographed in the Gulf of Mexico by an oil company, and they're quite blind.
Yeah.
But they have huge waving antennae and the most wicked-looking pincers you've ever seen, so I suspect they're ambush predators.
Yeah. Yeah, those pinchers look a lot like the, they remind me a lot of the mantis shrimp. Really long, sharp. Yeah.
Uh-huh.
Awesome. Hey, pilots, when we get up top on the sandy spot, can you find a shrimp and zoom in on it? I think I see 2 right now, but... Oh, yeah, thank you. I'm not sure if that's one or not.
Looks a bit big.
Looks a bit sargassum-y.
Sargass gummy.
I don't see any right now, but they were pretty abundant. Well, I wouldn't say abundant. They were pretty common earlier. There's some more bamboo corals coming into view. On the rocks, on the left. Right?
On the right.
On the right. My dyslexia is so brutal.
Scott reminds us that we did see a Chrysogorgia earlier with Chirostellid.
Oh, right. We were talking about...
But it wasn't well placed for collection. We were trying to... And we thought, we'll get another one.
And we haven't seen another one.
We haven't seen another one since that. That was really early on.
Yeah. We have like 5 more dives left, though, so they're everywhere.
Yeah.
We'll see more.
Maybe we'll see one on the midwater dive.
Mm-hmm. Yeah. At the bottom?
Yeah.
Ooh, something there. Can we look at that armed thing? Persingid, maybe?
Wow. Really long arms. Maybe it is.
Looks like a sea star or something. Oh, Persingid. Definitely different species than we saw earlier. This one has a lot of arms.
Right.
Maybe 12 arms, I'm counting, with a central that's very round. Really neat.
Yeah, he had said something about being interested in Persingids.
1, 2, 3, 4, 5, 6, 7, 8, 9. I think 12 armed.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 arms. I got the same.
The pink central disc. That's cool. So that's a different species than we saw the other day. I don't know the name of either of the species, but it definitely looks way different.
We saw one that was called Frayella.
That's right.
But yeah, this looks so much different than that. And I think we saw another.
Something in the water. Ooh.
Is it like...
Here we go. Amphipod.
Amphipod, I think.
It's the same, similar to the one we saw earlier that was really active. Nothing. Food. You can see how thin this ridge is. Maybe another yellow parameruseid over there.
Oh, we have a name for this Brisingid. This was Hymenodiscus.
Thank you.
That comes from Chris. Thank you, Chris Ma. Dr. Ma. My buddy back at the Smithsonian.
We have another Gorgonian at noon. Coming up.
Big.
Mm-hmm. Meter tall, maybe? Half meter tall? Yeah, bamboo coral. Yes, nodal branches. Very planar. There's a branching thing at 4 or 5 on those rocks. Could zoom in on that. I'm sorry, my dyslexia is terrible right now. 7 or 8. Yep. Dead coral. Stalk.
It's interesting. It had that
Had something growing on
Paramerissia growing on it.
I saw Hydromedusa in the water column.
Did you?
Yeah, you missed it.
Too much time in the chat. Who is reporting a fairly large lag.
Oh, really?
That's why Chris wasn't able to call in about the Brisingid.
There's some neat fans here.
Look
These are
at that.
different. Much bushier than the ones we had been seeing. Can we zoom in on those when you get closer? Or you feel comfortable? Another one of your Faria stems.
Yep.
Hexactinella.
The very long, thin.
Mm-hmm. Looks like stylaster on those. And then Bathopathes in the background, the yellow one.
Got all sorts of stuff going on in here.
Mm-hmm. Nodal branching on the bamboo coral. loads of critters. It looks like zoanthids on that branch up there.
That larger bamboo coral in the background is the one I was referring to as Isidella before. I don't know that that's what it is, but that's what I refer to it as. But there's a lot of it in here. I think that's probably the dominant morph or species that we're seeing today along this wall.
Quite a few of them. Little garden.
Yeah.
Fan garden.
Did we look in here at this
there's
brighter white sponge?
Yeah, white thing at 7. Now it's 6. Yeah. Might just be a rock, Alan. Oh, no.
Oh, it's another one of the...
Yeah, Holothurians.
Holothurians.
Look, you can see his little poo trail.
Yes, it's complete with poo. Really likes those pteropod shells. Thanks for that, Zoom.
Another bamboo coral, real fan-shaped. I think we saw this one already.
I think so as well. I think that must be 60, 70 or more centimeters across.
No worries.
Great. Quick pilot shift here. We've got about a little over a half hour left of bottom time. Out here on a ridge that we've been exploring. We're at about 2,350 meters, and we started, well, we've made 100 meters vertical. That's nice. Yeah. Seems like several of our dives we have not made the vertical distance that we had sort of hoped or planned.
Yeah,
for a variety of reasons. Sometimes it's difficult to make headway with the currents, or the collecting can take a long time.
Or headway with us.
Yes, or us just wanting to look at. Can you zoom on this? Can you zoom on that?
Yeah.
Can you zoom on this? And not. But
we got pretty far on this one.
Everyone is a little bit different. Some of them seem like they go by in no time, and others, and it's not just how many different things you see. Each one has a bit of a different feel to it.
They're all a blur at this point for me. I don't know what day of the week it is. I don't know how long I've been out here.
Who cares what day of the week is while we're out here?
I really don't care. It doesn't matter.
Yeah.
It doesn't matter because I'm not getting off the boat till I get off the boat. It's like I need to worry about it. I'm out here till I'm not out here.
Exactly.
Right? So why think about it?
Well, there are those Sundays.
Oh, we do have ice cream social Sunday, which is probably the only reason I have some idea of what day it is.
I missed the last Sunday.
I'll text you next time I see Sundays.
No, I was full.
Oh.
I had like a huge dinner that day, and I was like, I can't put a Sunday in me.
Food's been really good this cruise.
Yeah, the food has been delicious.
I usually luck out with that. That's good food out here. The big hats off to our chefs, man. They cook all day long. All day. To feed. How many people are on this boat? Do you know?
47.
47 people are on this boat. So we have 3 cooks who are chefs who cook for 47 people, 3 meals a day, plus desserts. Yeah, it's crazy.
Plus the overnight snacks.
Oh, plus overnight snacks, because we have a 24-hour ops on this ship. Ooh, that's beautiful.
Sweet. So we'll spend our last half hour continuing to explore this ridge. We'll make our final environmental DNA collection at the end.
I think we looked at this one.
I think we only zoomed from a far distance.
Oh, this is the one we saw from far away.
This is one of our best positioned ones.
This one's different from the ones that we were just looking at on the edge of the cliff. Those ones had the really 45-degree angle V-shaped branching, where this you can see it's much more curved main branches with branches coming off of that. So this is going to be a different species for sure.
Yeah. It's really organized.
Yeah, I suspect going to it, this is going to be the J clade, the quote-unquote Jasonisis clade.
And some,
We've got
comatulids?
Yeah, comatulid crinoids in there. Great. We're going to do a transect on this bamboo.
Definitely nodal branching.
Are these polyps that are coming off the main stem? And there's a little solitary coral,
no,
too.
I think those are aborted branches. Oh, but there are polyps just above it, if you mean on the upper right there.
Yeah.
Yeah. Yeah, you know, I think of these colonies, especially the really old ones, as constantly kind of replacing the tissue, and new polyps can be arising, and you can definitely see, like, just up a little bit here, there's a really small branch coming off where there's much thicker branches. And so clearly these are much more recent branching events compared to the original, let's say, scaffold of the colony itself. Really nice. So it's been down here for a very long period of time. It's been having different periods of growth. I wonder if it goes through growth spurts, you know. It had a growth long time ago, and then maybe the currents changed and didn't have as much food accessible, and then it goes through other more recent growth spurts. Just making all this up as I go.
Hey, Scott. Sorry, can you guys just twist a little to 3 o'clock? We've got this like, brambled, weird branching. So here's your anemones that you were talking about yesterday.
Yeah. I saw that as I was talking. I'm really glad you directed us back over there. Once again, this is more data. This is so useful. These observations that we're making, you get these ring anemones, and they get this name because the pedal disc, which is the part of the anemone that fastens to the substrate, is wrapped around the branch kind of like a ring. And the tentacles are facing away from us right now. And it seems when we get these, it induces something in the living tissue of the coral to then put out these wacky branches. So I'm not sure I understand it fully. You know, I think of it, I equate it to some kind of cancerous growth. You know, something goes wrong in the cells, and then other cells sort of go crazy in their growth. And that's kind of what we're seeing here repeated. Be something really interesting for study in the future. There's a nice face-on view of another one of these anemones. Good job, pilot and videographer. Très cool, très cool.
There's another
There's
one there.
another anemone.
Also, you know, we got to go back and look at the one that we saw, I don't know, several days ago. I think that was also on a J clade. One of my students is definitely going to start studying this.
The PhD project.
Look at that. That's nice.
Nice comatulid crinoid. You can see his
holding on tight.
Hold fast feet. They can get up and swim away. There's another anemone, too, in the foreground. Wrapped itself around.
I wonder if that comatulid has been there for a very long period of time. It looks like the tissue's been scraped off the skeleton.
Yeah. Especially right above it.
Yeah, well, if you got in such a nice spot.
Yeah, I wouldn't let go either. It's perfect.
Right,
high up
and
off the
there's
ground.
the question, right? Do they hang on when there's an open spot, or is it the action of them hanging on abrades the tissue?
Mm-hmm.
We need about 2,000 in situ cameras just to sit and record things for months on end at one place, so we can start to learn a little bit more about the ecology of the deep sea.
That one looks like it's lost most of its arms.
Yeah, it's all just Siri now.
Mm-hmm. One arm left.
That makes you wonder what comes along and nibbles on the crinoid.
Probably pretty crunchy. Something with teeth.
Nolan Barrett has made a comment in the chat room that I think is excellent, and he says that perhaps the reaction of these corals to the anemones is analogous to plant galls that are induced by wasps. There are many, many species of wasps that will lay their eggs in plant leaves, and the plant then forms cells around it to create this gall to try to close off the impact of where that parasitoid is.
Mm-hmm.
So Nolan, I agree. That's a really good analogy.
Another gorgonian here at 6 o'clock. Two of them, actually. Probably bamboo corals. Oh, another Faria stem.
Yep, that's nice.
Did that break up when you brought it in through the suction hose, or it maintain its...
I think it broke up. Yeah, didn't stay together, but that's okay. The imaging, maybe we can get another snap zoom on this one. This is such a well lined up.
There you go. He's
yeah,
so neat.
Please. Oh, you can see it's growing such a regular fashion. Oh, a nice associates with some amphipods on it. These little wings are alternating side to side to side to side. Really quite regular.
Yeah, I'm fascinated by the geometric regularity of these sponges, and it's just
weird.
Such a wonder to think about how supposedly simplistic an organism can build this complex, regular architecture.
There's a shrimp at 9 o'clock, little red guy. Now he's dead center. There you go. Hey, Mary, if you're still listening, this is one of the shrimps we've seen. She's on a bit of a delay, so if you guys could just hold there for a second. Really long legs.
That is great.
It's a nematocarcinid.
Nematocarcinid. Thank you.
There are 3 species. It's really hard to tell them apart from a photo.
Oh, yeah, I'm sure. Ooh.
There we go. Iridogorgia, right?
Mm-hmm.
Or...
Nice.
Yeah, I think so.
Prediction upheld.
Finally, you were saying you were hoping we were going to see these at the top.
Yeah, absolutely. Just because we saw that skeleton, that made most sense that it was an Iridogorgia. So, such beautiful corals. The world's largest known octocoral is an Iridogorgia at about 5 meters tall.
Wow.
This one isn't quite that tall, but it is certainly impressive. This is in the family Chrysogorgiidae, so it's a relative of the Chrysogorgia and Metallogorgia that we've been seeing previously. And characteristic of that family, the skeleton has a sort of golden iridescence, and the polyps are these little droplets on a very thin layer of tissue. So always delicate looking, very refined, and this is probably Iridogorgia magnispiralis. That magnispiralis species name gets the name from the very wide diameter to the coil of the axis.
Nice.
Goodness, I wonder how tall this is.
The lower branches as it grows. So the taller it gets, the fewer branches you see at the base. But when it would be a smaller colony, you would have branches, side branches at the base.
It's
I'm not
huge.
sure why that is, but that's what we observe.
Mm-hmm. I think almost 2 meters, I think.
I think it's more than 2 meters.
I think it's more than 2 meters? So that's a life. Are we on the left side of this mound now? Because we were hanging out on the right most of the time. I think we've swung out around to the other side of the south side. Okay.
Yeah, we've just sort of come, we're not looking at it the same angle as we have been. Really nice.
There's a species of shrimp that always lives on Iridogorgia, although I didn't see it on that one.
We can do a scan of it. Let's check. Hey guys, is there a shrimp on that spiral gorgonian we were just looking at? The spiral. If you see any shrimps on there,
I didn't see any.
No one saw any. Yeah. Okay. More bamboo corals. I see at least 20 of them.
Definitely good rock. Yeah,
Yep.
Bathopathes.
Mm-hmm. Yeah, those are different ophiuroids. We haven't seen many ophiuroids this dive.
Yeah, not until we got to the top, right?
Mm-hmm.
Are those little stylasterids?
Not sure.
Or
Looks like that coralliidy
they
thing
could be,
better.
yeah, small. Maybe that used to be a large colony of Corallidae, and it's just sort of
died
and hanging on a little bit.
Little piece left. There's also some ones that have broken off on the top or flopped over. And then one really...
Can we use the line again now?
Scott, what's the matter?
I said, can we use the line again now?
Use the line
I'm
again?
not dead yet.
Oh, yes, of course. You'll be dead in a moment.
Notice these guys are only on this one coral and not these other species.
Yeah, it's only on the dead one, it looks like.
Yeah, that's an interesting observation from Roland. You know, we don't usually see those Okeanos on bamboo corals or chrysogorgids. I've seen them on primnoids before, and I wonder if it has something to do with, I don't know if it's the polyps, the tissue, or the strength of the colony.
Yeah. Or some kind of chemical interaction. Right, some secondary metabolites that distract or keep them off.
Thank you. That was a good excursion.
Mm-hmm.
Good to get a look at that. It's another Iridogorgia.
Yep.
They are. I love their spiral morphology.
Yeah, really neat.
Whoa. Wow.
Really flexible.
Keeping my eye open for shrimp. I don't see any on this one yet.
Yeah, the shrimp that are associated with these Iridogorgia are typically pretty large. You wouldn't miss them.
Oh, okay. I was definitely looking for a small associate.
Yeah, no, you're not looking for a mysid this time.
Little tiny amphipod crawling on the stem.
Always, you look in the distance, and that's like, oh, look, there's even bigger stuff in the distance, and just when we're ready to take off from the bottom, we're going to see something really amazing. That's beautiful. Look at this. This must be a very stable part of the wall here.
Mm-hmm. Stable and gets good flow.
Yep. Yeah, to be a successful coral living for a very long period of time, you have to be lucky as a larva to settle and attach to the bottom before you know the full conditions, I suppose, and then not get buried in sediment while you're growing, and then have a constant source of food for the rest of your life. Because once you make that decision, there's no going back.
Yep.
No swimming away for you guys.
There's a pink thing.
What is that pink thing?
It does. Looks like a
bubble
gum.
That was a Paragorgiid.
Yeah. And there's another one, like orange. Oh, it's probably Bathopathes. Okay. Yeah, it looks like bubblegum coral. Really small guy. Yep. They're neat.
Wow. I like it. Hadn't seen that yet. Paragorgiidae, right?
Yeah, I'm pretty sure that's what this is. Part of me wants to say it's Swiftia, but no, it's Paragorgiidae.
Mm-hmm.
And those little small wart-like features you see may be the Siphonozoids. So they have dimorphic polyps on these colonies. So the smaller ones have no tentacles and just sort of channel water into the colony.
Mm-hmm.
Oh, that's cool.
I'm sure that's what this is.
Then we got a close-up of the bamboo it's sitting next to. The parts of the flesh gone. Maybe some stylaster on the stem. Man, definitely some zoanthids. See the yellow, the pinky colored.
Oh yeah.
Yeah, cup topped. Yep, yellow in the background.
Is that
Huh. It looks like it's coming off that main stalk.
that is interesting.
Another cornularid. This one's a yellow one.
Yeah.
A yellow cornularid. Wow.
Yeah. That's cool.
I'll just take this whole thing.
Yeah.
Yeah, we did have a request for trying to get some zoanthids. I don't see that there's any easily accessible here, and I know we don't have a lot of time, but it's too bad.
Yeah, if we were trying to get those, we'd take that entire bush, and we would have nowhere to put it, and B,
Just have to carry it back like that.
Yeah, flat Stanley it in front of this camera.
Oh, it's a jungle.
That's right. Forest of fans. All look like similar species. Another Bersingid maybe coming up.
Oh yeah. Right there in front of me.
Mm-hmm. That looks like the one we saw
That looks
earlier.
exactly the same.
Really circular central disc. Yep.
Hymenodiscus, I think.
Oh, we got a orange something or another. What is that? Sea star, maybe?
What?
Can we zoom in center right when you get a chance?
Curious what that is right below it, too. Oh, that's... What is it doing?
I don't know. It's
How
very
is-
rubbery. Oh, it might
I
be
think it's
eating.
a sea star eating on a coral.
Yeah, I think it's eating. You see it's the soft
Oh, yeah,
stomach
it's wrapped
poking
around
out?
there, yeah. I had trouble figuring out how it was,
Attached.
yes, how it was hanging out there, but
Little tube feeder out. Another Brazingid, I think. Oh, no, brittle stars. 2 brittle stars. 3 of them.
Are these the tiny, tiny white branching things, are those hydroids or are those...
I thought they were Stylaster. Can we zoom back in, guys?
Foraminifera?
They look like Stylaster to me, but yeah.
Do they? Okay.
yeah, I think Stylastarids.
Hmm.
They're so small.
Yeah, they're tiny.
I guess you got to start somewhere.
Guess this counts as a high diversity site.
Mm-hmm. Ooh, look at the Lepas, the barnacles.
Ooh.
Their tentacles out. Ooh, he's feeding. That one's feeding. Can we zoom in on him any closer? Or? There you go. So these are actually sessile crustaceans. Or, arthropoda. I don't think the crustaceans are arthropods.
Crustaceans.
I think crustaceans is suborder, isn't it? Superfamily, it's one of those.
Subclass.
Subclass. Oh, it was close.
These belong to a family called the Scalpelidae.
Scalpelid. Nice.
Belong to a family called the Scalpelidae.
Okay. There's a bright red pink thing.
This is really great, and this is again what happened when we got up towards the end of the dive in 2017 or 18, when we were on that ridge a little bit deeper and a little bit farther to the northwest, and we found these really large bamboo corals and some pretty high density with lots of diversity when you look close, like we just were. So, really nice to see this here.
Nice. So we're back on a little bit of a sandy patch. Ooh, and a lot more gorgs over on the other side.
Yep, another little promontory edge covered with fans.
Probably bamboo corals is my guess. They're all bent kind of the same way.
A couple corallids here.
Mm-hmm.
Bright white really stands out.
There's this really interesting tree in California at the beach, and they all kind of grow like with the wind.
Mm-hmm.
So all of them, you look at them, and they're like up on the rocks and stuff, and they kind of grow bent over like this. They remind me of that.
Quite familiar with
Yeah.
that scene out on the coast of
Mm-hmm.
California. Really.
Neat. They look like a jade plant or jasper or something, like you usually buy them bonsai.
Like juniper?
Juniper. That's the word I'm looking for. They look like a giant juniper.
Yeah.
What's that brown thing at 9? Like a brown ball. Right there, yeah.
Have you found a sponge?
I may have found a sponge. I don't know from this far away. Ooh. Boop. Sponge.
Yeah. For sure. Beautiful demo sponge. Could see right through into its spongy interior there.
Okay, we're good, thank you. Looks like Cleona or something, but I don't know if Cleonia's down this deep. Another stalked crinoid. Mm-hmm. Yeah, we're facing north, so probably coming up from the ledge towards the east. Might be a current that comes up that way. More Bathopathes. More Paramyrcia, the yellow.
Mm-hmm. Just keep thinking forests of grandeur.
Mm-hmm. No, that's fine. There's some more Zoanthids in there. Talk to the bottom.
The yellow guys?
Mm-hmm.
Wow. We share the planet with this.
I know. That's so crazy.
It's just...
There may be a Chrysogorgia in
I was
there a little bit
thinking that,
further
too.
ahead at about 3 o'clock
Yeah.
past the fifth
Yep, yellow.
bamboo coral.
Yep, right there.
Yeah.
Oh, that's a good description, apparently. No, I'm wrong. That's not a Chrysogorgia, is it?
I don't know. It's very fine. Very bushy.
It is, actually.
I don't see any arthropods on it.
Got to be a Chrysogorgia. The color of the skeleton
Mm-hmm.
and the angularity of the branching, but I don't recognize that open pattern. It's some kind of very big and open Chrysogorgia.
Mm-hmm.
The genus Chrysogorgia. Very interesting.
Oh, there is an
there is
arthropod
one.
on the backside.
Can we zoom in on the crab right there at the bottom? Do you see him?
He's hiding in
Yeah,
there.
there we go.
Also,
That guy.
maybe an egg. That might be an octopus egg, Dumbo octopus egg on the Chrysogorgia.
Where? This white thing?
The white thing.
Oh, nice.
The white thing with the yellowish. They have like a mud coating over the egg.
Oh,
That
neat.
might be what that is. A little bit too far.
Yeah, I think we're coming off bottom in like 2 minutes.
Really, really
Yeah, so it's
beautiful.
Too late to collect.
I can't believe... So that's
Chrysogorgia with a Dumbo octopus egg.
Now,
It could. I mean, it could be.
Dumbo or that group?
Like Chrysogorgia, and I mean, there you could see why it gets the name Chrysogorgiidae, which translates as the golden coral. So you could really see that reflecting nicely, that gold iridescence.
Mm-hmm.
Hey, Okeanos.
Hey.
Mike Becky on here.
Hey, Mike.
Hey, I think Scott's right. I was thinking the same thing, but was a little hesitant to commit to it. I do think that those were cirrate octopod eggs.
Oh, nice.
Looks like there was at least 2 of them on that Chrysogorgid.
So I know the octopus that I'm familiar with, I guess the more shallow water ones, they usually crawl themselves into a hole and lay their eggs and then babysit their eggs and don't eat. And then after their eggs are hatched, they die. Is that the same kind of life cycle choice? Well, I guess not really choice, but life cycle that this Sarianthid have? Yeah, he only has 2 eggs here. And so, there's no mother care.
Well, the Dumbo octopods lay their eggs on deep sea corals. We know that. And they lay them individually and then go away. Now, we don't know a whole lot about the life cycle. So we don't know if they die after they spawn or whether they might continue spawning for a prolonged period of time. I know that some of the ones I've dissected have had eggs of varying stages of development, which sort of indicates that they probably keep spawning for a long time, but they don't regress into a post-spawning stage and then mature more eggs for a second batch later on.
Okay.
So as we zoom in, are there 4 in there?
I don't know how much lag I've got. I don't know how much lag I have, but I see we've zoomed back in again.
Yeah, I think there's 4. I see 4 of these round tan balls. There's one at the top.
At least.
Yeah, I'm not so sure about the one that's off to the right a little bit that looks
spiky.
I don't know, sort of spiky.
The upper one might be complete. It doesn't look open, but the third one in the middle, that looks already hatched maybe, but I'm not sure about that second one, Mike, that they're zooming in on now.
Yeah, I don't know either. But one thing that Okeanos discovered when they were in the Pacific was that these coverings are really tough, and I always wondered how the embryo could get out when it hatched, how it could get out through the covering, and it turns out the egg swells and actually bursts the covering when the embryo is ready to hatch.
Cool.
So they have like a trap door that pops open on the end. And I don't see that on any of these. But I don't know what else these could be other than cirrate eggs, and they are consistent with cirrate eggs. Did you get a measurement on them?
We can. Can you guys paint these with lasers real quick? Yeah. Maybe 2 to 3 centimeters, if that. What do you think?
Yeah, that's okay.
Yeah, couple, 2 to 3.
Yeah, that's consistent with a cirrate egg.
Okay.
Scott.
So unless Scott knows something else they could be, that's the best guess as to what we're looking at.
Cool.
Yeah, the only other thing might cross my mind was an Ascothoracicin, but I think they're too big. They're far more consistent with the eggs.
So that one's hatched.
You should send them to.
Get a good.
You should send them to Abby because she's doing a manuscript on cirrate eggs.
Absolutely, it's sitting here on my desk.
Well, that looks open as well, right?
Mm-hmm.
That one looks like it's open. It looks like like I said, they pop open at the end.
Yep.
That one looks like it's been hatched.
I bet you baby Dumbo octopus are so cute.
I bet you as well. Who knows? Maybe we saw that one down the hill.
It takes them a while to grow into their fins.
That shrimp.
Yeah, what do you got, Aunt Mary? What do you think?
The shrimp is Bathypalamonella for sure.
Bathypalamonella.
I don't suppose you could collect one of those so we could get DNA on it.
I would love to, but we are like coming off the bottom like right now. Otherwise, I would have grabbed that whole thing.
We don't know the identity of any Syrian octopod eggs.
Oh, really?
Then a guy has done some. There's a guy in Germany that's been doing micro CT on them and trying to identify the embryos based on adult characters.
Oh, yes, please, in this skin bottle before we take off bottom.
Mike, we were on a cruise in 2005, and we brought one up, and it hatched on board in a bowl.
Was it adorable?
Yeah, I saw the paper that was published about that.
Yeah,
I actually reviewed it.
What Tim did with the tissue, so there may be a chance to do DNA at least on that one. That was on a Chrysogorgia as well.
That's going to be
yeah, I don't know whether they sequenced that one or not.
Yeah.
Did they get
That's
it?
interesting to think about. If you can get Tim's attention long enough to find out.
Yeah, good luck.
All right. Thanks for everybody's help today.
Well,
that was awesome.
I guess we say goodbye.
Yeah, we're coming off bottom.
Mike.
Thanks so much for tuning in, everybody. It's really helpful to us.
Thanks,
and
Mary.
especially when I'm out of my depth range that I don't know stuff.
Well, that's
typically
every depth range for me.
Yeah. So, yeah, thanks for chiming in, everybody. That was really helpful.
Everybody's out of their depth range down here.
Right? That makes me feel good.
Yeah, so that was a really nice ridge dive.
It was. It was awesome.
We made it all the way to 2,350 meters, so we basically made about 100 meters of vertical relief along the edge of this reef.
Yeah.
Edge of this ridge, excuse me. It was kind of reef-like at places.
Yeah, right. We are going to be diving tomorrow on a sinkhole on the Portales Terrace. If you want to chime in on that dive, feel free to stay on the line. We're going to have our post-dive call or pre-dive call for tomorrow right after probably the next 15 minutes or 10 minutes or so. So please hang on the line. We'll be back shortly. But just want to do a dive recap?
Sure.
Go ahead, Alan.
Well, let's see. Well, we started off with a very sandy bottom, and we wound up encountering some holothuroids, which we continued to encounter in the sandy spots. That was
the purple one, right?
Purple, and then also a tongue-shaped sort of pinky one. Both of the family Synallactidae. And we encountered not abundant, but hexactinellid sponges throughout. We saw some Hyalinema, that's a stalk sponge, and a Pharonema, which is an allied taxon that grows on the bottom with hair-like structures in the sandy bottoms. And then we were able to collect a really interesting-looking hexactinellid Phareidae, the family Phareidae, that grew like a long sort of backbone, very thin and narrow.
Yeah, really geometric.
Very geometric.
We saw quite a few of those. I would say call those common up on the top part of the ridge.
Absolutely. And then we saw lots of different kinds of octocorals throughout. So we saw Chrysogorgia at the beginning and the end, and then also we saw Iridogorgia at the end, a huge colony that must have been 2-3 meters tall. Of course, we saw the Magna Pinna squid float by. Fortunately, we caught just the hint of the tentacles and decided to look up to see what we could see, and then we got great footage of this large big fin squid. We saw primnoids, bamboo corals. What else?
That holothurian that likes to coat itself with seashells.
Yeah, Olopholinus. We saw that.
Lots of Bathopathes. Let's see, we sampled 5 water samples, which is what we usually do. I sampled a bamboo coral, a yellow paramyriceid, a rock, the Faraday that Alan was just talking about, and then another paramyriceid with the associate of an ophiuroid or Astroshima. So 10 samples today.
Yeah, and we saw a couple different of these octocorals that have tubes, that grow out of tubes, these cornularids, including one really striking yellow one towards the end of the dive. for some reason, they're very intriguing to me. Probably because they're hydroid-like.
Yeah.
But I really like those guys.
They're super weird.
We saw not a whole lot of fish, but we got some great footage of a tripod fish, and then also an ally to a tripod fish that lives in the sand and has very rudimentary eyes.
Oh yeah,
but
like
a big like plate on its forehead for sensing light.
Brazilian stars.
Shouldn't call them rudimentary.
Yeah, they're probably super advanced.
Exactly.
Probably the opposite of that.
Complex, but different than sort of normal.
Well adapted to this depth and light, light accumulation.
Yeah, and then we saw Nematocaris shrimp. Nematocarcinus, not a Nematocaris. And a lot of great things.
Came up the wall. That was really neat. The wall looked really cool. Lots of black rock, and then on top is mostly this sediment. It's pretty thick enough to have fauna in it because you see lots of bioturbation. I think that's probably about it.
Yeah, and then just that we saw a real concentration of the large fans on the
edge.
On the one edge of this.
Yeah.
On the current side,
yeah.
Not surprisingly, and so that's sort of as we would expect it.
Yeah,
so
totally.
It was a really great dive.
Yeah, it was a good dive. So,
hey everyone.
Hey.
Let's check back in at 4:00 to do the dive planning call for tomorrow.
Okay.
We have a really nice shelf edge on the Florida Strait to work with tomorrow. It'll be a nice waypoint between the dive today and getting up to the Blake Plateau in a couple days. So, let's take a break, and we'll see you at 1600, 4 o'clock.
All right. Thanks, Matt. All right, so we're signing out. If you, let me get my paperwork here, my notes. You can follow NOAA Ocean Exploration on Twitter at Ocean Explorer, on Facebook at Ocean Exploration Research, or on Instagram at NOAA Ocean Exploration. You can follow me, Stephanie, on Twitter at Deep Sea Nerdy on Twitter, or at Deep Sea Nerd on Facebook or Instagram. And you can follow Alan on Twitter at Collins_NOAA SI, or on Facebook at Alan Collins Zoologist. Please tune in tomorrow. Should have another cool dive tomorrow. If all goes well. Of course, everything out here is pending weather and current, all that stuff. But thanks for tuning in. This is Windows to the Deep 2021 and NOAA Ocean Exploration. Have a good afternoon, everybody.
Thank you. Bye.
Thank you.